Today let's start a new chapter which comes in 11th NCERT, unit number 4, plant physiology, chapter number 13. It is photosynthesis, right? So this is very important chapter in terms of board also, in terms of NEET also. So let us try to understand what is photosynthesis in single shot. In one shot we are going to complete the chapter, okay? So now if we talk what is this process and what's the significance of this process, means all animals including humans, we depend on food on plants, right? So from where do we get our food? From plants only. The animals which eat the plant directly are called herbivores. The animals which eat plants indirectly are called carnivores. Means we can tell that all animals including human beings depend on plants for their food. Let's write down. All animals including humans we depend on plants for our food, right? Now the question is, from where will plants get food then? From where will we get food means from plants. Now the question is, from where will plants get the food? From where will plants get the food? Now this is a question. Now the answer is, plant will not get the food, plant will manufacture the food. The green plants, they manufacture the food by a process called photosynthesis, right? So we can tell the green plants manufacture food by a process. What is the process called? By a process called photosynthesis. Now come on, what is synthesis? Synthesis means synthesizing a compound using photon energy, sunlight energy is called photosynthesis. Now then, if we have to see photosynthesis, what it is, means if I am writing PS means photosynthesis. So photosynthesis is synthesizing food, right? So photosynthesis means it's a process by which plants can prepare the food. Means it's a process of synthesizing food. Now food means it is sugar. Sugar means it is organic material, right? Any synthetic process is catabolic process or anabolic process. Means I'm asking you, photosynthesis is catabolic process or anabolic process? It is synthetic process. It is synthesizing sugar. Means it is an anabolic process. So we can tell photosynthesis is anabolic process. It is anabolic process, right? So all the anabolic process require energy. Here the energy is sunlight. So since sunlight, the physical source of energy, is converted to chemical source of energy called ATP and NADPH2 or sugar, we also call photosynthesis as a physico-chemical process. Photosynthesis is an anabolic process. We can also tell photosynthesis is physico-chemical process. What do you mean by physico-chemical process? Because physical light, sunlight is used, it is physical chemical process. So sunlight the plant will absorb, but it cannot directly use the sunlight. So that's the reason it converts sunlight into chemical energy called ATP and NADPH2. So it converts the sunlight energy into chemical energy. Now what is that chemical energy? Direct energy is ATP, reduced form of energy is NADPH2. Since it is absorbing the sunlight, is a physical process. Since it's converting the physical energy to chemical energy, it's called physico-chemical process. Now, if we have to show the equation of photosynthesis, we will write down like this, right? 6 carbon dioxide molecules plus 12 water molecules in the presence of sunlight. Who will absorb sunlight? Pigments. What is a major pigment? Chlorophyll. Where is the chlorophyll present? In the chloroplast. So, the raw materials, the inorganic materials are carbon dioxide and water. In the presence of sunlight, the pigment of the plant, chlorophyll, what it will do? It will reduce carbon dioxide to carbohydrate. So it will reduce carbon dioxide to carbohydrate. What is carbohydrate? Glucose, C6H12O6. So C is there, O is also there, hydrogen came extra. Means hydrogen is moving from here to here. Hydrogen is moving from here to here. Means addition of hydrogen is called reduction. When you remove hydrogen from here, oxygen will be left out. 6O are left out. 12 O's are left out, we can write it as 6O2 and 6H2O molecule. This is a complete balanced equation of photosynthesis. The inorganic materials are carbon dioxide and water, and the organic material what we get, the main product of photosynthesis is sugar, glucose. The byproduct is oxygen. Plant will release oxygen, with that oxygen only we are doing respiration. Okay, so here what happens, carbon dioxide is reduced to carbohydrate, water is oxidized to oxygen. So carbon dioxide is reduced to glucose, carbohydrate, whereas water is oxidized, right? From water you are removing hydrogen, so it is oxidized to what? It's oxidized to oxygen. Since reduction reaction and oxidation reaction both are involved, we also call photosynthesis redox process. So we call it's an anabolic process, it's a physicochemical process. Based on this equation, we can also tell photosynthesis is redox process. Reduction is also there, oxidation is also there, so we can also call it as redox process. Okay, so then when will plant do photosynthesis? In the presence of sunlight. Means all animals are depending on plants for their food, and plant prepares its food by sunlight. So we can tell that sunlight is the only source of energy for the entire earth. So we can tell sunlight is the only source of energy for all the living organisms on the earth. Sunlight is the only source of energy for all living organisms on the earth. Sunlight is the only source of energy for all the living organisms on earth. And who can trap the sunlight? Means we cannot trap the sunlight, only plants can trap the sunlight. That's why plants are the only producers. Plants are the only producers which can trap the light and convert that sunlight into chemical energy called ATP and NADPH2. So I think you understood so far what we have discussed. Take a screenshot, we'll continue. Right, so we are telling sunlight is the only source of energy for all the living organisms on earth, and plants are the only producers, we are telling. Now photosynthesis is important for two reasons. What are the two reasons? Means it gives food for all the living organisms, that is one reason. Along with the food is releasing oxygen through which we are living, we are respiring, that is the second reason. So in NCERT, in the first para, the author says that photosynthesis is important for two reasons. One, it's the primary source of food for all the animals on the earth, right? So let's write down. Photosynthesis is important for two reasons. What are the two reasons? One reason, it's the primary source of food for all the animals on the earth. It is the primary source of food for whom? For all the animals on earth. This is one reason which we tell photosynthesis is important. And the second thing is, along with the food, it's releasing oxygen also. It releases oxygen through which animals are respiring, they're breathing and they're surviving. It releases, photosynthesis releases oxygen. This is the second reason why we tell photosynthesis is important. Then since it is 13th chapter, so next topic is 13.1. The author will ask us, what do we know about photosynthesis? Now the next topic for us is 13.1. What do we know about photosynthesis? What do we know about photosynthesis? Means we are going to talk about certain experiments which are related to photosynthesis here in this topic also, in the next topic also. Now the first experiment what we tell here is variegated leaf experiment. What is the first experiment? So let's write down experiment number 1. It is called variegated leaf experiment. Now what do you mean by variegated leaf experiment? Usually the leaf will be green in color, but there are some leaves where other colors also will be there, right? There are some leaves where we find patches of other colors also, right? We find patches of other colors also, whereas the main color will be the green color only. So all this is green color. So if it is having different colors like that, like green color it is having, white color it is having, red color it is having, such a leaf is called variegated leaf. Now in variegated leaf experiment, what is the significance of variegated leaf experiment means? Now we know that plant appear green in color because of the presence of chlorophyll pigment. Now pigment means it absorbs sunlight. So this is a pigment which absorbs sunlight and it converts the sunlight energy into ATP and NADPH2, uses that energy to make glucose. So in variegated leaf experiment, what we have done is we have taken a plant and we expose this plant to sunlight. So the green part is also exposed, non-green part is also exposed. So then we know that only the green part will absorb sunlight. Only the green part will absorb sunlight. They absorb sunlight, that means they are doing photosynthesis. They will do photosynthesis. So how do you know that they are photosynthesis? If they do photosynthesis, they prepare glucose which gets converted to starch. Right? So how can you check the presence of starch? Iodine solution. So we are telling that this green colored pigment, so will absorb the sunlight. It will absorb the sunlight, it will do photosynthesis and it manufactures starch. We can check the presence of starch by adding iodine solution. So when you add iodine solution, what happened means the red colored part will not do photosynthesis because it's not trapping the light. At red colored part, there's no change in iodine. Whereas the green colored part, starch is there, it has done photosynthesis, starch is there, so it will do photosynthesis and the starch gets converted to blue color. So we can see that the leaf turned blue, means there's a change in iodine color. Only when starch is there, then only it reacts with iodine and it changes the color of the iodine. So where is starch there? Where the green color is there. So that means this says that the green part of the plant, in the presence of light will do photosynthesis. Right? So this is a significance of variegated leaf experiment. Understood, children? This is a significance of variegated leaf experiment. So starch reacts with iodine. And changes the color and gives blue color. So now if you have to give the conclusion, what is the inference? What is the inference of variegated leaf experiment? Means the green parts of the plant. The green parts of the plant will prepare photosynthesis, will do photosynthesis and prepare starch. The green parts of plant absorbs sunlight and it will do photosynthesis. So this is the significance of the first experiment, which is called variegated leaf experiment. So we told photosynthesis is important for two reasons. It prepares the food and it releases the oxygen. Then what do we know? 13.1 topic when we come, the first experiment is variegated leaf experiment. In variegated leaf experiment, so we came to know that the green color parts of the plant will have chlorophyll that absorbs sunlight and it prepares starch. That starch, when we react with iodine solution, it changes the color. Whereas the non-green parts of the plant, now they did not do photosynthesis. Since they did not do photosynthesis, starch will not be there. When you add iodine, there will not be any change in color. So take a screenshot. We'll go with experiment number 2. Right? Now let's talk about the second experiment. First experiment is variegated leaf experiment. The next experiment is called as Moll's half leaf experiment. Through variegated leaf experiment, we came to know that the green parts will absorb the light, will do photosynthesis. So what is experiment number 1? It is called variegated leaf experiment. What is our conclusion here? The green parts of plant absorb sunlight and they will do photosynthesis. Now let's go on to the second experiment. The second experiment is called as Moll's half leaf experiment. A scientist called Moll, what did he do? He has conducted an experiment in which half of the leaf is kept inside the glass jar. Now see, this much portion of the leaf is kept inside a glass bottle. This much portion of the leaf is kept inside a glass bottle. Inside the glass bottle there is cotton which is dipped in KOH solution. Now this is cotton which is dipped in KOH solution. Now what KOH solution will do? It will trap the carbon dioxide. Whatever carbon dioxide which is there inside the bottle is taken up by the KOH solution. So there is no carbon dioxide inside. Okay. So they have subjected this plant to the sunlight. Means sunlight is taken by this portion of the leaf also, as well as sunlight is taken by this portion of leaf also. Light is there, and through the xylem water is also coming. Water is also coming. So sunlight is there, water is also coming, carbon dioxide is exposed here. Atmospheric carbon dioxide is there for this half of the leaf, whereas the remaining half of the leaf where KOH is absorbed carbon dioxide, no carbon dioxide. This portion of the leaf, carbon dioxide is not there. This portion of the leaf, carbon dioxide is there. So when carbon dioxide is not there, carbohydrate will not be formed. So that means this experiment says that carbon dioxide is required for photosynthesis. So this portion of the leaf which is exposed to atmospheric carbon dioxide, which is taking the sunlight, which is taking the water, so what it will do? It will do photosynthesis and it will prepare starch. How do you know that the starch has been prepared? It changes the iodine color. Iodine solution changes to blue. Whereas this portion of the leaf, water is going, light is also there, but since there is no carbon dioxide, they don't do photosynthesis. No photosynthesis, then no starch. When there is no starch, no change in color of iodine. No color change. So this is called as Moll's half-leaf experiment. And what's the inference of Moll's half-leaf experiment? That carbon dioxide is required for photosynthesis. Carbon dioxide is required for photosynthesis. Take a screenshot. We'll talk about the third experiment. Alright. So can I erase this? Now let's talk about the third experiment. Now that came in 13.2 topic. Early experiments this author has given us. So experiment number 3, it was conducted in the year 1772 by Joseph Priestley. It's called bell jar experiment. It's called bell jar experiment. Now experiment number 3, which is conducted in the year 1772, it's called as Joseph Priestley's bell jar experiment. Now what did Priestley do? So in NCERT they have given us 4 pictures they have given, right? The first picture, what did they give us? In the first picture they showed us that the candle is turned on, and next one they showed us that mice is alive, right? So candle is turned on and mice is alive, which is kept inside the bell jar. It's kept inside the bell jar. So this they have given NCERT as B. Now what happens after some time? What happens after some time? Now after some time the mice got suffocated and it died. After some time the mice got suffocated and it died, and they saw that the candle also turned off. It's not lighting, right? The candle also turned off. This is experiment number B. Now the third setup what he has taken is, he has taken a mint plant inside the jar. He took the candle. Again he brought a live mice. We'll keep the mice in between. He kept a live mice, and this time he brought a mint plant also. He brought a mint plant also. This is mint plant and this is setup number C. Now what happens after some time? What happens after some time? Now it is a glass jar. Glass jar means sunlight will come. Right? So now then what happens? Plant is taking the sunlight. Excuse me. Plant is taking the sunlight, and then the mice is doing breathing, right? It is doing breathing and it releases CO2, and the burning candle also the fumes. Right, when he kept a mint plant, what is his inference? His inference is when plant is not there, the mice died and the candle turned off. But when plant is there, he told that whatever the breathing animal and the burning candle has removed from the air have been added by the plant, he told. He did not tell that the plant will release oxygen and the mice will release carbon dioxide. It was not his inference. That was given by the next scientist, John Ingenhousz. He just told that whatever the burning candle and the mice will consume are restored by the plant. So, okay, so what is his inference? He told whatever the burning candle and the breathing mice removed, whatever they removed are restored by the plant, he told. Means, what did they remove? They removed oxygen. What did plant restore? Plant added oxygen by doing photosynthesis. This is what he told. But then he don't know that the plant is releasing oxygen and animals are releasing carbon dioxide. That he don't know. Even though he discovered oxygen, the same scientist in the year 1774, he discovered oxygen in the atmosphere. He discovered oxygen, but he don't know that the plants are releasing oxygen. So if they ask you what is the inference of bell jar experiment, you can tell that when he took this setup, after some time, what happened? The mice got suffocated and it died because it's a closed compartment, and the candle also turned off because the air has been converted into, in the air, what happened? Oxygen got exhausted. Now when he kept a mint plant, after some time, the candle is still on, the mice is still alive because the plant is restoring the components what they are taking. This is his inference. Okay, now take a screenshot. We'll go to the fourth experiment, which is conducted by John Ingenhousz. Now third experiment got over, Joseph Priestley's bell jar experiment, and we are seeing the inference also. Now experiment number 4 was conducted by, in the year 1779, his name is John Ingenhousz. He did an experiment. He took the same setup, the bell jar setup he took. What is that? Mint plant, candle and the mice he took. And he kept the this thing in the presence of light and in the absence of light. He did the same experiment what Priestley has done, but he did it in 2 different setups. Once he kept the setup in the presence of light, the other setup he kept it in dark. So when he kept it in dark, even though the plant is there, when you keep it in dark, even though the plant is there, so now come on, think what will happen. When sunlight is not there, plant, can it do photosynthesis? It cannot do photosynthesis. If it is not doing photosynthesis, will it release oxygen? No. So then if oxygen is not there, then what will happen to the mice and the candle? So when the plant is not restoring the components back, since it is not doing photosynthesis, since it is not getting sunlight, so then what happens? As usual, the mice will die. And as usual, the candle also will turn off. The candle turned off when in dark. Okay, whereas the same setup, if you take in the presence of light, the same setup when you take in the presence of light, when sunlight is coming, then what plant will do? Plant will take carbon dioxide and it releases oxygen, and using this oxygen, what happens to the mice? Mice will be alive. Plant is releasing that oxygen is taken by the mice. See, mice is laughing, it's very much alive, and the burning candle also will be on because oxygen is there. Burning candle requires oxygen, right? So it will also be on. He told that it's not just plant presence is required, plant in the presence of light only will do this. So what is the inference of Jan Ingenhousz? Let us write on. He told that whatever the mice and candle foul the air will be purified by plant. Will be purified by plant. He told that plant will purify the air only in the presence of sunlight. They'll purify the air in presence of sunlight. We have seen that in presence of sunlight only it can do that. Okay, but here also he don't know it is oxygen which the plant is releasing. He conducted one more experiment, elegant experiment, to tell that it is oxygen. Okay, so then again he did another experiment. This is extension of Priestley's experiment. Other one is, we call it as elegant experiment. In this elegant experiment he has taken an aquatic plant. Now he has taken an aquatic plant and he exposed the aquatic plant to bright sunlight. So when you take an aquatic plant and when you expose, aquatic plant means it will be there inside the water, and when you expose the plant to bright sunlight, then it will be doing photosynthesis. When it will do photosynthesis, it will prepare food also, and it will release oxygen bubbles also. So it releases oxygen bubbles into the atmosphere, into the water. So these are small bubbles. He noticed some small bubbles, and when he checked what is there inside the bubbles, he came to know that it is oxygen. Then he came to know that plants when they are exposed to bright sunlight, then they are doing photosynthesis. They prepare their food starch. They also release small bubbles which contain oxygen, and this oxygen only is making the mice to be alive and is making the candle to turn on. It is this person who told that in photosynthesis oxygen comes as a byproduct. Right, children? We have seen 4 experiments. The first one is variegated leaf experiment. The second one is Moll's half leaf experiment. The third one is Bell jar experiment, which is conducted in the year 72. And the fourth one is Jan Ingenhousz 2 experiments he did. One is extension of the Bell jar experiment. The other one is elegant experiment, who proves that it is oxygen what the plants are releasing, which is essential for the life to sustain. Take a screenshot. We'll go with the 5th experiment. Now the next experiment was conducted by Julius von Sachs in the year 1854. This scientist name also comes in mineral nutrition chapter. There he did it in 1860. So this he did in 1854 only. He tells that plant will do photosynthesis and it will prepare starch or it will prepare glucose. That glucose is stored inside the chloroplast itself, and chloroplast are there inside the mesophyll cells. Okay, so he tells the product of photosynthesis is glucose or starch. Let's go to experiment number 5 in the year 1854. His name is Julius von Sachs. Right? Jan Ingenhousz, what did he tell? Like plants do photosynthesis and they release oxygen, he told. Now, what did Julius von Sachs told? He told the product of photosynthesis is starch or glucose. He told the product of photosynthesis is glucose or starch. He also told that, Julius von Sachs also told that, like plant absorbs the sunlight. Plant absorbs sunlight. Now, who will absorb the sunlight? Sunlight is absorbed by chlorophyll pigment. And where is chlorophyll pigment? He told chlorophyll pigment is there in a special organelle. Pigment is there in special organelle. The organelle is called as chloroplast. The organelle is called chloroplast, and in the chloroplast it will do photosynthesis and it prepares starch, and the starch is stored in the chloroplast only. Starch is stored in chloroplast only as starch grains. So this much information he gave. He told that photosynthesis is light dependent process. Sachs told that photosynthesis is light dependent process. Plant absorbs light. Who will absorb light? The pigment. What is a pigment? Chlorophyll. Where is the pigment? In the chloroplast. Where is chloroplast? In the mesophyll cell of the leaf. It will do photosynthesis by absorbing the light, and it will prepare starch or glucose. It stores the starch inside the chloroplast itself as the starch grains. So this much information we came to know through Julius von Sachs experiment. Okay, now let's go to the next experiment. What is that? Experiment number 6, or we can tell experiment number 6 was conducted by T.W. Engelmann. Right, so experiment number 6 was conducted by T.W. Engelmann. It's in the year 1888. So it is in the year 1888. T.W. Engelmann has drawn the first action spectrum of the plant. So he drawed the first action spectrum. Now, how to draw the first action spectrum means, so he told that the chlorophyll will absorb the light. Now, we know that the light what we see is a visible light. So, visible light is a spectrum. V, I, B, G, Y, O, R. Violet, indigo, blue, green, red, orange. All these wavelengths are there. So, what did he do means, he has taken the wavelength of light on the x-axis. The wavelength of light is V, I, B, G, Y, O, R. Right, it ranges from 400 nanometers to 700 nanometers. So the visible range of wavelength, it's a spectrum. It starts from violet, indigo, blue, green, yellow, orange and red. He has taken the wavelength of light on x-axis, and on y-axis he is checking the rate of photosynthesis. The rate of photosynthesis he is checking by measuring the oxygen evolution, by measuring the amount of oxygen which is evolved. So if you have to measure the amount of oxygen evolved, so it should be an aquatic setup, right? In the air, if it goes means it disperses, oxygen disperses, but here we can get collected in the form of oxygen bubbles. So we can collect those bubbles. So he's checking the rate of photosynthesis by the amount of oxygen evolved. So this person, Engelmann, he has taken a green algae. He has taken a green alga. What is that called? Cladophora. He has taken a green algal plant, it's called as Cladophora. He exposed the plant to sunlight. Right? He exposed the plant to sunlight. He exposed the plant to sunlight at violet wavelength, at indigo wavelength, at blue wavelength, at green wavelength, yellow wavelength, orange wavelength and red wavelength. Okay. And then he wants to see at which wavelength it will do more photosynthesis. So he saw that one at blue wavelength and another at red wavelength it will do more photosynthesis. How did he check photosynthesis? Oxygen release. So it will release more oxygen where? At blue wavelength. Again it will release more oxygen where? Again at red wavelength. So how can you see this oxygen evolution? He has taken a suspension of aerobic bacteria. He has taken a suspension of aerobic bacteria. Aerobic bacteria require oxygen for their growth. Now, if the plant is doing more photosynthesis, it will release more oxygen. If it releases more oxygen, bacteria will grow more. If it releases more oxygen, then the bacteria are growing nicely. He saw that these are the bacteria. He saw that the bacteria is growing at blue wavelength and red wavelength. So the rate of oxygen evolution, if you start like this, it will be more for blue. Yeah, here it's more. And it will be least for green, and again it should be more for red wavelength also. Again for red wavelength also. At blue wavelength, the oxygen evolution is more, right? And at red wavelength also, oxygen evolution is more. More oxygen evolved. So thus the first action spectrum is being described by T.W. Engelmann in the year 1888. So he has taken a green algae called Cladophora. He was checking that green algae Cladophora absorbs light at which wavelength and it will do photosynthesis at which wavelength, right? It absorbs more at blue wavelength and red wavelength. The more the absorption of light energy, so then it can convert more ATP and it can make more NADPH2. It can do more photosynthesis. It can release more oxygen. So using that oxygen, bacteria will grow more. So this was the conclusion given by Engelmann. So for this, what you need to do, so you need to take a light source. we need to take a light source and we need to split the light into individual wavelengths. We need to split the light at violet wavelength, at indigo wavelength, at blue wavelength, at green wavelength, at yellow wavelength, orange wavelength and red wavelength, right? So the instrument which is used to convert the polychromatic light, this light is called as polychromatic light. Polychromatic light means different wavelengths are there. When you pass that light to a prism, now what is prism? Prism is called as monochromator. What will monochromator do? It will split the light into individual wavelengths. So it is splitting the light into violet, indigo, blue, green, yellow, orange and red. So then he saw at blue wavelength they are growing more, at red wavelength they are growing more. This is a first action spectrum. Take a screenshot. We'll go with experiment number 7. Right? So experiment number 6 was conducted in the year 1888 by T.W.Engelmann. What is that? Action spectrum. Now by the mid of 19th century we can tell, by the mid of 19th century, empirical equation of photosynthesis has been drawn. Empirical equation of photosynthesis has been given. Children, in this way only you have to read NCERT. In this way only we have to read NCERT. The line to line of NCERT we should understand thoroughly. So what is an empirical equation means? It is a rough equation. It's not a complete balanced equation. What is the empirical equation we got in the 19th century means? They told carbon dioxide and water are required by the plant in the presence of light. It will convert carbon dioxide into carbohydrate and it releases oxygen. This is an empirical equation which we could give by the end of 19th century, in the mid of 19th century. Then we can go with experiment number 7. Experiment number 7 was conducted by a microbiologist. His name is Carl von Niel, or we can tell Carolus von Niel. He conducted that experiment. We don't know the year. Carolus von Niel. He is a microbiologist. He conducted experiments on green sulfur bacteria and purple sulfur bacteria. He conducted experiments on green sulfur bacteria and purple sulfur bacteria. And what did he tell means? He tells that for doing photosynthesis, water is not always required. He tells if any compound that gets oxidized is sufficient. He took an equation like carbon dioxide plus 2 H2A molecules. You need not take water molecules. If you take any compound like H2A, then also the hydrogen comes here and you will get carbohydrate. And these two A's will be released out and a water molecule also comes out. He tells that photosynthesis can be done without evolution of oxygen. Here oxygen is not evolved, right? Oxygen is not coming. Why? Because water is not taken. So indirectly he tells that the oxygen which is evolved in photosynthesis comes from water but not from carbon dioxide. He also tells that photosynthesis can be done without evolving oxygen by certain bacteria called green sulfur bacteria and purple sulfur bacteria. These bacteria are obligate anaerobes. They are obligate anaerobes, means they die in the presence of oxygen. So that's why they don't release oxygen. Without releasing oxygen, they do photosynthesis and they survive. So this is an experiment. What is this called? Milestone Discovery. Once in NEET examination they asked, what is a milestone discovery? It is Carolus von Niel experiment where we came to know that photosynthesis happens without oxygen evolution, right? So he tells that the oxygen which is evolved in photosynthesis comes from water. But we need a scientific proof, right? So then Ruben and Kamen has conducted radioactivity experiments, radio-labeled experiments in the year 1941. In the year 1941, Ruben and Kamen, what did they conduct? They conducted radio labeling experiments to prove that the oxygen comes from water but not from carbon dioxide. So how cleverly they have taken, means they have taken carbon dioxide with radio labeled O16, they have taken water molecule with radio labeled O18, 6 CO2, 12 H2O gives rise to C6H12O6 plus 6 O2 plus 6 water molecules. They saw this oxygen is from 16A, radio labeled 16A, radio labeled 18A. This oxygen is from 16A, 18A. Product water is 16A, 18A. They came to know that in glucose the oxygen is there is with O16 only, and in oxygen the O2 is from O18, and the product water's oxygen is from CO2. It is from CO2, right? So they came to know that the oxygen which is evolved in photosynthesis is water's oxygen. If you take water, you will get oxygen as a byproduct. Take a screenshot, we will continue. Now we finished these experiments, right? Now the next topic is 13.3 in NCERT. Where does photosynthesis takes place? That is our next topic. Hope you understood all the experiments. So any details if you need, you can comment them in the comment box. Let's go to the next topic. That's 13.3. It is where does photosynthesis takes place. So we know that photosynthesis is done by the leaf, right? So let's go to the next topic. 13.3, it is where does photosynthesis takes place. We know that photosynthesis takes place at the leaf. In the leaf, in the which cells means it is in the mesophyll cells of the leaf. In the mesophyll cells, in which organelle means we also know it is chloroplast. In the chloroplast, where means it has a pigment. The pigment is chlorophyll. The main pigment is chlorophyll A molecule. The abundant pigment which is present in the nature is chlorophyll A, which is called its reaction center. Here the photosynthesis takes place. So we came across, right? We are telling photosynthesis, where does this take place? The author has asked a question. The answer, it takes place in the leaf. In the leaf, where it is? If we see the structure of the leaf, we tell this is an adaxial epidermis and this is an abaxial epidermis. This is a vascular bundle, and then we draw the cells here. So these are parenchyma cells, mesophyll cells, right? So I'm drawing palisade parenchyma here, and if you have to show spongy parenchyma which are irregular, so these are spongy parenchymatous cells. And here we have phloem, this is phloem, and this is xylem, vascular bundle, which helps in conduction of food and water. So these are the mesophyll cells. This is spongy mesophyll, spongy parenchyma, mesophyll cells, and this is palisade parenchyma. So we are telling this is anatomy of the leaf. Now on the mesophyll cells, photosynthesis happens. In the mesophyll cells, what are there? Chloroplasts are there. So I am drawing the disc-like structures, so like this. So these represents chloroplast. Now the chloroplasts are semi-autonomous organelles. Where are they present? Means they are present in the leaf, in the mesophyll cells. Now these mesophyll cells which are having chloroplast, which have a pigment called chlorophyll, they absorb the sunlight and they do photosynthesis. So this is a place where photosynthesis occurs. So we concluded that the light is absorbed by chlorophyll, right? So we are telling that the light is absorbed by the pigment called chlorophyll. So to absorb the light, the chlorophyll or the chloroplast should be hiding inside, or they should be located parallel to the mesophyll walls. So I drawed all the chloroplasts parallel to the walls only, peripheral to the walls only, so that they can trap as much as light as possible. So we came to know that the chlorophyll takes place, the photosynthesis takes place in the pigment chlorophyll. Where are chlorophyll or where are chloroplasts present? They are present in the mesophyll cells. We are telling that the chloroplast align, right? The chloroplast align peripheral to the walls of the mesophyll cells. They align peripheral to the walls of the mesophyll cells. Why? To trap the sunlight so that they can do photosynthesis more efficiently. In NCERT, the author has asked when are the chloroplasts parallel and when are they perpendicular. So they are asking us, if you take this as a wall and this as an incident sunlight, so when we have to draw the chloroplast cells like this, one chloroplast and another chloroplast, they are asking us when are the chloroplasts parallel to the walls and when are they perpendicular to the walls. So this is parallel wall, right? They are asking us when the chloroplast will be like this, parallel to the walls, and when they are perpendicular, they are asking perpendicular to the wall means this way. When are the chloroplasts perpendicular, they are asking, right? So when the light intensity is low, so at low light intensity, to trap as much as light possible, the chloroplast will be parallel to the walls. Whereas at high light intensities, the pigment undergoes destruction. The pigment gets denatured, it gets destroyed. So it has to hide itself from the high light intensity to prevent photooxidation. Then they will move off. They are perpendicular to the walls. Here they are. So we can write down at very high light intensities, right? At high light intensities. At high light intensities, they are perpendicular. At low light intensities, they are parallel. So we are telling they are parallel to the walls. They are parallel to the walls at low light intensity and they are perpendicular to the walls at high light intensity. So when they are perpendicular, they prevent photooxidation. Photooxidation means it is denaturation of the pigment. Understood till here, children? Now, if you take 1 square millimeter of the leaf surface, if you take 1 square millimeter of the leaf surface, you know, 500,000 chloroplasts will be there. That much chloroplasts are there. 500,000 chloroplasts are there in the leaf. Now, if we have to see the dimension, what is the length of the chloroplast and what is the width of the chloroplast? If we have to discuss what is the length of the chloroplast and what is the width of the chloroplast, means the length is 5 to 10 micrometers and the width is 2 to 4 micrometers. The length of the chloroplast is 5 to 10 micrometers and the width is 2 to 4 micrometers. Take a screenshot, we will continue. Right? So now, if we have to draw the structure of chloroplast and see what is the shape, the typical shape of the plant chloroplast, it is disc-shaped structure and it has two membranes, outer membrane and inner membrane, and both the membranes are the same like mitochondria, they don't have any folds. The inner membrane will enclose the protein part which is called as stroma. So come on, let us draw the structure of the chloroplast. Inner membrane, this is outer membrane. Now this is the length we are telling, which is 5 to 10 micrometers in size, and this is a width we are telling, which is 2 to 4 micrometers, right? And this is the outer membrane of the chloroplast, and this becomes the inner membrane of the chloroplast. The space between outer membrane and inner membrane is called as periplastidal space. This space which is here is called as periplastidal space, and the inner membrane is enclosing this part. All this part is called a stroma, protein rich stroma. Now this region is called as stroma. Since chloroplast is having two membranes, it's a double membrane structure, it's a disc shaped structure. So we have seen the length also, we have seen the width also. Now inside the chloroplast there are certain membranous sac like structures which are called as thylakoids. So here is one thylakoid, and these thylakoids will pile up one on top of each other to like a coins, how they are stocking. Now this complete structure is called as granum. Now one is called as thylakoid. It is called grana thylakoid. Now one, when I draw it, it is called as grana thylakoids. If they are piling up, then that is called granum. Not one granums are there, so many granums are there in each chloroplast. So I'm drawing just one more grana here, and I'm drawing one more grana here, right? So all these granums together, 1, 2, 3 granums together is called as grana. The plural form is grana, granum is singular. And if we draw many granums, so I draw 3 granums, many granums is called as grana, and these grana are interconnected by stroma thylakoids. Now this grana is interconnected to another grana by stroma thylakoid or stroma lamellae. This is also connected to this by stroma lamellae. So what is this? This is called as stroma lamellae, or we can call it as stroma thylakoid, or we can call it as fret membrane. Okay, so granum is connected to another granum by stroma lamellae. Stroma lamellae can connect the granum to the membranes also. It's connecting it to the inner membrane also. Since it is autonomous organelle, it has its own circular double-stranded DNA. So here I'm drawing its double-stranded circular DNA, and it manufactures starch here, and the starch is seen as starch granules. In NCERT, they showed us the starch granules in this manner, right? These are amylose granules, so they are called starch granules, and it has its own double-stranded circular DNA. Since it's having its own DNA, it is autonomous, and it contains ribosomes also. So these are 70S ribosomes of the chloroplast. These are 70S ribosomes of the chloroplast. And Blackman is a scientist who told photosynthesis is biphasic. So Blackman told that photosynthesis has a light phase and it has a dark phase. Blackman told photosynthesis has light phase and dark phase. Light phase happens in the grana, dark phase happens in the stroma. All this is called stroma, right? Now there is division of labor inside the chloroplast. Light reaction in the grana, dark reaction in the stroma. Where will light reaction happen? Light reaction in the grana. Where will dark reaction happen? In the stroma. So did you understand the structure of the chloroplast? Take a screenshot, we'll continue. Right, when we have to differentiate what is light reaction, what is dark reaction, let us see that. Blackman is a scientist who told that there are light reactions and dark reactions in photosynthesis. Let us see what do you mean by light reaction and what do you mean by dark reaction. Just now we made out one point that light reaction occurs in the grana of the chloroplast. It occurs in grana. And we saw that dark reaction occurs in stroma. It occurs in stroma. This is one difference. And when we have to talk about the next difference between light reaction and dark reaction. In light reaction, what happens, what is there on the granum thylakoid membrane? Since pigments are there, chlorophyll A, chlorophyll B, xanthophyll, carotenoid pigments are there. These pigments, they trap the sunlight. They convert the photon energy, quantum energy, sunlight energy into chemical energies called ATP and NADPH2. Means in light reaction, sunlight will be absorbed and that light energy is converted to chemical energy. So what can we write in light reaction? In light reaction, photon energy is absorbed Who will absorb? Is absorbed by the pigments. They will absorb the light energy and they convert the light energy into chemical energy. What is the chemical energy? Direct chemical energy is ATP, reduced chemical energy is NADPH2. Okay, so this is one point which happens in the light reaction. This is one point which happens in the light absorption will happen and light energy is converted to chemical energy, it will happen. Okay, and that happens in the grana. So that is why we can tell that light reaction, as we can also call it as a photochemical reaction. Photochemical reaction. Photon energy is converted to chemical energy, that's why it's called photochemical reaction. You can also call this as light dependent reaction. Light is required for that, sunlight energy is required, so it's called light dependent reaction. Now, four things are happening. The first one is absorption of the light. The second one is conversion of light energy into chemical energy called ATP and NADPH2. Right? So, when they convert the light into these things, what happens? Photosystems will be there. The pigments, where are they present? They are present in the reaction centers called photosystems. These photosystems, when they absorb the sunlight energy, they get excited. When they get excited, the electrons will jump to higher orbitals. They lose their electrons because electrons are jumping and they are going off. When the electrons go back, when they go off, you have to give the electrons back to it. So when they absorb photosystem, when it absorbs sunlight energy, it gets excited. During the excited state, it loses electrons and it becomes photosystem plus. Now you need to give electrons back to this photosystem to make it normal. That means from where will the photosystem gets the electrons? Means with the splitting of water. What is this? Splitting of water. Splitting of water in the presence of sunlight, we can also call it as photolysis of water. In mineral nutrition chapter, you told manganese and chlorine are required for photolysis of water. Photolysis of water, where does it happens? In the light reaction. So when you break the water molecule, you will get 2 protons, you will get 2 electrons and you will get oxygen. So these electrons are taken by photosystem to become normal again. So then oxygen is also evolved. So 4 things I told in light reaction. The first one I told, light energy is absorbed. The second one, light energy is absorbed to converted to chemical energy. The third one, photolysis of water. And the fourth one, oxygen is evolved. So these 4 things will happen and it is called as photochemical reaction. It's called light dependent reaction. It's called energy accumulation reaction. It happens in the grana. Now what will happen in the stroma? In the stroma, dark reaction will happen. So what happens in the dark reaction means energy got accumulated. This energy is used to convert carbon dioxide to carbohydrate. For example, if I had to buy a car, first I need to accumulate that much amount of money, then I can go to the showroom and I can purchase the car, right? So in the same manner, if it has to synthesize glucose, it needs energy. That energy is prepared here. When that energy is ready, then it will go to the dark reaction and it will convert carbon dioxide to carbohydrate in the stroma. So what happens in dark reaction? We can tell in dark reaction, carbon dioxide is converted to carbohydrate. Carbon dioxide is converted to carbohydrate or it is converted to sugar, we can tell. Since carbon dioxide, which is a gas which is reduced to carbohydrate, we can also call it as carbon dioxide fixation phase. We can also call this phase as carbon dioxide fixation phase, since the carbon dioxide is converted to carbohydrate. It's fixed to carbohydrate. And conversion of carbon dioxide to carbohydrate, it is an enzyme mediated process. It's a synthetic process. So we can also call it as biosynthetic phase. This is called light dependent phase, photochemical phase. This is called as carbon dioxide fixation phase, or we can also call it as biosynthetic phase. Since sugar is getting synthesized here, we can also call it as biosynthetic phase. So don't think that dark reaction happens in the dark. Since they call it as light reaction, the alternate word they used is dark reaction. But dark reaction also occurs in the daytime only. Dark reaction also occurs in daytime only. Just as an alternative, they have used the word called dark. Don't think that it happens at the night time and this happens in the daytime. So light reaction, we told it is light dependent reaction because it needs light energy to convert the light energy into chemical energy. Here light is not required. Here light is not required. So we can tell dark reaction is light independent reaction. Light is not required, so it is light independent reaction, but it depends upon the products of the light reaction. What are the products of the light reaction? ATP and NADPH2. Energy it require to join carbon dioxide molecules to make carbohydrate. So we can tell dark reaction is light independent reaction. Light is not required, right? So it is light independent, but it depends on the products of light reaction. It depends on the products of light reaction. So what are the names for dark reaction we told? It is carbon dioxide fixation phase, it's called carbon phase, it's called biosynthetic phase, and it occurs in the daytime. It's called carbon phase also. It occurs in the daytime only. When the energy gets accumulated, it switches off from light reaction to dark reaction, and using that energy it will make carbohydrate. When the energy gets exhausted, again it will come back to light reaction, again it starts accumulating these things. So this is the information about light reaction and dark reaction. And who is more affected? Who is more affected when the temperature becomes more? Means dark reaction is more affected when the temperature becomes more, because their enzymes are there, and these enzymes are sensitive to temperature. If you increase the temperature, the enzymes will not work. So we can tell like dark reaction is more sensitive to temperature than light reaction. In light reaction, we told ATP is synthesized, right? So if the ATP is synthesized in a cyclic manner, what is it called? So we can tell in light reaction there are 2 steps. In dark reaction also there are 2 steps. So in light reaction we are synthesizing ATP. Synthesizing ATP in a cyclic manner is called cyclic photophosphorylation. It is called cyclic photophosphorylation. Phosphorylation means making ATP. Making ATP by using photon energy, sunlight energy, is called photophosphorylation. Synthesizing ATP in a cyclic manner is called cyclic photophosphorylation. So in light reaction we can synthesize ATP in a cycle. It's called cyclic photophosphorylation. There's another way where we synthesize ATP in a non-cyclic manner. Here also ATP will be made, but not in a cyclic manner. It's a Zscheme. So this synthesis of ATP is called non-cyclic photophosphorylation. What is this scheme called? It's called non-cyclic. It is Z, no, means it is non-cyclic. It is non-cyclic. Sunlight is only used. Phosphorylation. This is called non-cyclic photophosphorylation. The ATP which is synthesized in the light reaction can be either by cyclic manner or non-cyclic manner. The carbon dioxide which is synthesized in the dark reaction, so it can be again of 2 types. One is called a C3 cycle. The other one is called a C4 cycle. In some plants, before making a 6-carbon sugar, it makes a C3 carbon, then it makes a 6,6 carbon. In some other plants, first it will make a 4-carbon compound, then it will make a 6-carbon compound. So the carbon dioxide converting into carbohydrate in dark reaction, if it goes by a 3-carbon compound, it's called C3 cycle, discovered by Calvin. So this is also called Calvin cycle. Okay, and C4 cycle was seen by Hatch and Slack. So it's called Hatch-Slack cycle. So these are the differences between light reaction and dark reaction. Take a screenshot, we'll continue. So the next topic is how many pigments are involved in photosynthesis. We'll discuss that also. Are you thinking that there's only chlorophyll pigment because the plants and the leaves appear green in color? Then it's wrong. So there are 4 types of pigments which are present in the plant. The main pigment is chlorophyll only. It's chlorophyll A and chlorophyll B. Along with chlorophyll A and chlorophyll B, it has carotenoids also and it has xanthophyll also. How do you came to know that? So our next topic is 13.4. How many pigments are there? How many pigments are involved in photosynthesis? 13.4. How many pigments are involved in photosynthesis? For that we need to do a technique which is called paper chromatography. Let's go to do paper chromatography. Children, graphy means separation. Chromato means separation of colored pigments. Paper, separation of colored pigments on the paper. Separation of colored pigments on the paper is called as paper chromatography. For that, we'll take a paper first. Yeah. I have taken a Whatman filter paper. Okay. And then I've taken a Whatman filter paper. I took a green colored leaf. I pasted it, and that paste we are adding it here. Here we are adding that green colored paste, and then we are keeping it inside the chromatographic chamber, in which a beaker will be there. Now this beaker has some buffer solution. Some buffer solution will be there. Some mobile ions will be there. So this is a buffer solution. There we have kept it. Okay, and then on that we'll keep a glass rod and we will roll the paper. We'll roll the paper. Now what happens? Because of capillarity action, the water or the buffer which is present here starts moving up. When the water or the buffer starts moving up, then the pigments also start moving. And when the paper becomes wet here, that means the separation has been completed. We have to stop the process. And then when we dry it, so there are 4 patches it seems. So one patch here they observed, another patch here they observed, third patch here they observed, and the fourth patch here they observed. So when they have done paper chromatography, they came to know that there are 4 patches. So one is chlorophyll B, the other one is chlorophyll A, other one is xanthophyll and carotenoids. When they saw how many pigments are there, when they have done paper chromatography, they saw that 4 pigments got separated. Now those 4 pigments are, one is chlorophyll B, next one is chlorophyll A, next one is xanthophyll, next one is carotenoids. These 4 pigments are separated. Now chlorophyll B, in which color we can see? Carotenoids, which color we can see means chlorophyll A, it is bright green color. So in which color it is? It is bright green. Or we can tell chlorophyll A will be in blue-green color. That is why when we see the plants, they are bright green or they are blue-green, because chlorophyll A is a major pigment. Whereas chlorophyll B, if you see, it will be light green in color. Chlorophyll B color, it will be light green in color. Whereas if we go for xanthophyll, xanthophyll is in yellow color. And carotenoids are orange colored. So these are the 4 pigments. Now you can ask, so when there are yellow color also, orange color also, why do we see only green color? Means the green color pigment is abundant and it masks these carotenoids and xanthophyll. So that's why we can't see that yellow color and orange color pigments. If you take papaya, papaya is orange in color because it contains carotenes. And next what we can take capsicum also, all these red and orange color fruits and vegetables, they have carotenoids more. And the leaf which is falling down, which will be yellow in color, it contains xanthophyll. Okay. So this is paper chromatography. Through paper chromatography, we came to know that there are 4 pigments: chlorophyll A, chlorophyll B, carotenoids and xanthophylls, right? Take a screenshot, we'll continue. Now, what did you understand by pigment? Pigment is a substance which absorbs light. So when we have done pigment, we came to know that paper chromatography, there are 4 pigments. Now the question what the author is asking is, what is a pigment? Pigment means it is a substance. It is a substance which absorbs light. Which absorbs light. right? So now for that we need to do absorption spectrum to see like which pigment is absorbing light at which wavelength. So let's do absorption spectrum now. Absorption spectrum. Now we need to take the wavelength on x-axis. So starting from 400 to 700 nanometers means it starts from violet, VIBGYOR. Individual wavelengths, visible wavelength we have taken on x-axis, and since we are measuring the absorption, we need to take the absorption range on the y-axis. So we can tell it is the absorption of light by the pigment. Absorption of light by the pigment. We are taking it on y-axis. We are taking it on y-axis. Now we came to know that the pigments will absorb light at which wavelengths. First, if you have to start with chlorophyll A molecule, chlorophyll A, it is in blue-green, no? We'll use blue color. At blue wavelength it has to give more, red wavelength it has to give more. So at blue wavelength it is giving more, and at red wavelength also it absorbs more. So through this we came to know that this is chlorophyll A molecule. Chlorophyll A molecule is absorbing light more at blue wavelength, is absorbing light more at red wavelength, and is absorbing least at green wavelength. It's absorbing least at green wavelength. And now let's go for chlorophyll B molecule. Chlorophyll B also, it matches the same. It also absorbs at blue wavelength only, and it also absorbs at red wavelength. Chlorophyll B is in light green color, no? So that's why I used green color. So chlorophyll B is also absorbing at blue wavelength only. It is also absorbing at green wavelength only. And carotenoids and xanthophylls are there, right? For that we need to use yellow color. I don't have yellow. We are using black color. Carotenoids, they absorb at blue and green. So carotenoids will give peaks like this. So these are carotenoids. So where are carotenoids absorbing? Carotenoids are absorbing at blue and green. They are absorbing at blue and green. Chlorophyll B is absorbing at blue and red. Chlorophyll A is also absorbing at blue and red. What is the spectrum called? This spectrum is called absorption spectrum, since you have taken the absorption of light by the pigment on y-axis. Now when we do action spectrum, Engelmann has done action spectrum. So when we do action spectrum, we have done action spectrum for chlorophyll A molecule only because it is a main pigment, it is a reaction center. So when we do absorption spectrum or action spectrum, action spectrum for chlorophyll A, then what are we taking on y-axis? Here we take the same thing, VIBGYOR, wavelength of light. Here we'll take oxygen evolution, right? Rate of photosynthesis you are checking by the amount of oxygen which is released out. Here for chlorophyll A, when we take, it is at blue wavelength it will evolve more oxygen, and at red wavelength it will evolve more oxygen. At blue wavelength it has evolved more oxygen, at red wavelength it has evolved more oxygen. So this is action spectrum which is done by T.W.Engelmann using Cladophora aerobic bacteria. So this is action spectrum. The third picture in NCERT, they overlapped absorption spectrum and action spectrum. So when you overlap action spectrum and absorption spectrum for chlorophyll A molecule, the action is like this, right? The action is like this, and the absorption, if we have to see, the absorption is like this. So if you see, this is absorption and this is action. Are they overlapping? Yes, they are overlapping at the peaks. Both of them are overlapping at the peak here. Both of them are overlapping at the peak here. Roughly they are overlapping, which tells that more the absorption, more the action. If it absorbs more sunlight, then it can trap more sunlight. It can convert more sunlight into more ATP and more NADPH2. So through this, we can tell that more the absorption, more the action. What do you mean by action? More the photosynthesis. Take a screenshot. Now through this we came to know that the main pigment is chlorophyll A, That's why we have taken action absorption for chlorophyll A, and there are other pigments like chlorophyll A, chlorophyll B, carotenoids and xanthophylls. So what will these pigments do? See, we have seen that chlorophyll A will absorb at blue and red only, whereas the other pigments are absorbing at all other wavelengths, right? So we have seen what is the abundant pigment in the plant. The answer goes as chlorophyll A. What is the reaction center? The answer goes chlorophyll A. Okay, so who will do photosynthesis? Chlorophyll A. What's the abundant pigment? Chlorophyll A. But this chlorophyll A, which is in the reaction center, will absorb only at blue wavelength, and it will absorb only at red wavelength. That means it will waste all other wavelengths like violet, indigo, yellow, orange. It will waste. So then comes the accessory pigments. So this is the chlorophyll A is a main pigment, we can tell, or we can tell this is essential pigment or reaction center molecule, whereas the all other pigments, what are they, children? Chlorophyll B, xanthophylls and carotenoids. What will these pigments do? All these pigments, they act as accessory pigments. They act as accessory pigments. These accessory pigments can absorb light at all different wavelengths. They can absorb at violet also, indigo also, blue also, yellow also, orange also, red also. They absorb at all different wavelengths, and they slowly pass on that light to the reaction center. So what are the roles of the accessory pigment? If you ask only chlorophyll A, it absorbs at its choosy, it absorbs at blue and red only. But the accessory pigments absorbs all the wavelengths of light, means it's not wasting the light energy. Their roles are to absorb all wavelengths of light, not to waste the light. That's the first thing. And the second thing is, so this chlorophyll A is choosy also, it is sensitive also. If the chlorophyll A molecule is subjected to continuous wavelength of light, then it undergoes photooxidation, it undergoes destruction. So these accessory pigments, they will mask the chlorophyll A molecule. If this is chlorophyll A molecule, all the accessory pigments, they will mask, they'll completely cover the chlorophyll A molecule so that it's not exposed too much to the sunlight. So what they will do, first thing is they're absorbing all the wavelengths of the light, and the second thing is they prevent chlorophyll A from photooxidation. They are protecting the chlorophyll A from photooxidation. Photooxidation is also called solarization. If solarization happens, chlorophyll A molecule is destroyed, it is lost, it is denatured. The accessory pigments are masking the chlorophyll A molecule. That's the second thing. And if accessory pigments are not there, it will absorb at 2 wavelengths only, then the yield will be less. Since it is absorbing all different wavelengths, they enhance the yield of photosynthesis. They increase the yield of photosynthesis by absorbing all ranges of wavelength. Understood? So this is a role of accessory pigments and this is the role of main pigment. So our next topic is what is light reaction? We will see. Take a screenshot. So now we'll see what happens in light reaction. Now light reaction happens in the grana. In light reaction, light will be absorbed and that light energy is converted to chemical energy. Since photon energy is converted to chemical energy, it is called photochemical reaction. Where does light reaction happen? Light reaction happens in the grana, right? Now there are four main events in the light reaction. The first one is absorbing sunlight. Second one is converting the sunlight energy into ATP and NADPH2. And when the light is absorbed, then the photosystem gets excited. It will lose the electrons. We have to give back the electrons to the photosystem. So we'll split the water molecule and we will give the electrons to photosystem. For that, the third event, splitting of water. And when you split water into protons, electrons and oxygen, the oxygen will be evolved. That's the fourth event. These four events, 1, 2, 3, 4 events happen in light reaction. Now light reaction, we are telling that happens in the grana. The grana thylakoid contains pigments. Pigments will absorb light. One pigment, two pigments, or hundreds of pigments are there. Hundreds of pigments are there. When hundreds of pigments are there, they need to be organized into complexes. They are called as light harvesting complexes. There are two types of light harvesting complexes. Light harvesting complex 1 and light harvesting complex 2, which are part of photosystems. Photosystem 1 and photosystem 2. That's what the author has given here. Light is absorbed by the pigment, and the pigments are organized into two discrete light harvesting complexes called as LHC1 and LHC2, which are parts of photosystem 1 and photosystem 2. I told you each light harvesting complex is made up of hundreds of pigments which are bound to protein. Each light harvesting complex approximately contains 200 to 300 pigments bound to protein molecules. If we have to see, this is a light harvesting complex. This we are taking it as light harvesting complex 1. This light harvesting complex 1 is made up of several types of proteins. And again, if you have to draw light harvesting complex 2, this is light harvesting complex 2, which is also made up of several hundreds of proteins. And then light harvesting complex is part of photosystem 1, and this is part of photosystem 2. This is LHC1 and this is LHC2, we are telling. Okay, so it consists of several hundreds of pigments. Like, what are the pigments? Means it can be chlorophyll B, it can be carotene, it can be xanthophyll. It is chlorophyll B, it is carotene, it is xanthophyll. Now these light harvesting complexes which are bound to proteins, they act like antennae. They will tell, they act like, they form light harvesting systems called as antennae. They are antennae. Why are they called as antennae? Means they receive the light at different wavelengths. So it receives the light at one wavelength, it receives the light at one wavelength, it receives the light at one wavelength. It takes the light and it passes on the light to the reaction center. Now if you see, like photosystem 1 has reaction center which absorbs light at 700 nanometers. This is a reaction center of photosystem 1, which has a special chlorophyll A molecule which is called as P700. Whereas the photosystem 2 has also a reaction center which also has a special chlorophyll A molecule which is called as P680. This special chlorophyll A molecule present in photosystem 1 absorbs at 700 nanometers only. But chlorophyll B, xanthophyll and carotenoids, which are called as antennae, which are called as light harvesting complexes, they absorb at different wavelengths and they pass on that wavelength of light to the reaction center. They are called shield molecule, they are called shield pigments, they are called accessory pigments, but we cannot call them as reaction center because they are not doing the reaction, they are just passing on to the reaction center molecule, right? So now this reaction center molecule receives the energy from chlorophyll A, from carotenoids, from xanthophyll. They will receive the energy, and this P700, after taking the energy, it gets excited. It gets excited. The excited state of P, we can write it as, yes, this is the excited state. During excited state, it will lose electrons. During excited state, when the photosystem gets excited, it loses the electrons. Now these electrons jump out of this photosystem or P700 and they are accepted by a primary electron acceptor. So the electrons from P700 are taken by a primary electron acceptor, right? So like that, the electron transport starts here. In the same manner, here also chlorophyll B will be there, carotene will be there, xanthophyll will be there. They will trap light at different different wavelengths, and they pass on that wavelength of light to the reaction center molecule. The reaction center molecule absorbs light at 680 nanometers red, and this absorbs at far red, and this also gets excited. This excited state is called as P680. In excited state, this will also lose electrons. Now the electrons from here also are accepted by primary electron acceptor. They are accepted by primary electron acceptor, and the electron transport starts, right? So that's what they have given here. Like each photosystem, this is a photosystem, has all the molecules except one special chlorophyll A. Special chlorophyll A is part of reaction center, whereas all other molecules, all other pigments act like antennae. They make the photosynthesis more efficiently by absorbing different wavelengths of light, because reaction center absorbs at red wavelength of light only. A single chlorophyll A molecule will make the reaction center. Photosystem 1 chlorophyll A absorbs at 700, photosystem 2 chlorophyll A absorbs at 680 nanometers. Take a screenshot, then we will talk about the electron transport. Right. Now, as I told you, in light reaction, there are two types of phosphorylations. One is non-cyclic, other one is cyclic. First, let us talk about non-cyclic electron transport. Now, the electrons which are released from this photosystem, how are they transported? How are they transported? That we need to see now. And who explained this scheme of non-cyclic electron transport? Means Hill and Bendal explained it. So we can tell non-cyclic electron transport chain. ETS, electron transport chain. Non-cyclic electron transport chain was explained by Hill and Benda. Right? Non-cyclic electron transport chain was explained by Hill and Bendal, and it shows a scheme which is called Z scheme. The movement of electrons will follow a particular scheme which is called as Z scheme, where both the photosystems will operate. Where will non-cyclic electron transport chain occur? Means it occurs on the grana thylakoid. It occurs on the grana thylakoid membrane. How many photosystems are involved in non-cyclic? Means both the photosystems. Photosystem 2 it will start, then it will move on to photosystem 1. Okay, it occurs on the grana thylakoid membrane. Now let us start the non-cyclic electron transport. So we will start with photosystem 2. We'll start with photosystem 2. Photosystem 2 has a pigment. What is that? Photosystem 2 absorbs at 680 nanometers. Photosystem 2 absorbs light at 680 nanometers. Now, 680 nanometers means it absorbs red wavelength of light. After it absorbs red wavelength of light, what happens? It gets excited. Now, when it gets excited, the excited state, we can symbolize it as P680 star. Now, in excited state, the electrons will jump to higher orbitals. It loses the electrons, and then it becomes photosystem plus. It has lost the electrons, and these electrons are picked by. These electrons are picked by, we are telling, a primary electron acceptor. They will ask you in NEET examinations, who is a primary electron acceptor for photosystem 2? Non-cyclic electron transport starts with photosystem 2. Now photosystem 2, which has a reaction center, which has a chlorophyll A molecule at 680 nanometers, it absorbs red wavelength, gets excited, loses the electrons. Those electrons are accepted by primary electron acceptor. The primary electron acceptor here, it is pheophytin. What is it? It is pheophytin. Now, movement of electrons from photosystem to pheophytin, primary electron acceptor, it is up the hill. It is uphill transport. Now this pheophytin will transport the electrons to plastoquinone. It is transporting the electrons to plastoquinone. Now this plastoquinone is not an electron transporter, it is a proton transporter. Since it is a proton transporter, along with these electrons, it takes protons also from the stroma. The pheophytin, which is a primary electron acceptor, which has received the electrons from photosystem 2, will transfer the electrons to plastoquinone. This plastoquinone, where it is located on the membrane only, which is facing towards the stroma. This plastoquinone is not an electron carrier, it is a proton carrier. Now because it's a proton carrier, along with the electrons, it takes protons also. And plastoquinone, after taking electrons and protons, it gets reduced, it becomes plastoquinol. And then this plastoquinone will transport the electrons to electron transport system. It will transport electrons to electron transport system they have given. If we have to see what components are there in electron transport system, means cytochrome molecules are there. If we have to list down the cytochrome molecules which are present here, so now we have to tell that the plastoquinone will transfer electrons to cytochrome B6 and F. It transports electrons to cytochrome B6 and F. From cytochrome B6 and F, further the electrons will go to plastocyanin. They will go to another molecule called plastocyanin, which is a mobile carrier. So the electrons of photosystem 2 are now taken by pheophytin, then by plastoquinone, then by cytochrome B6, then by plastocyanin. Now meanwhile, here we have photosystem 1, which has P700, which will also absorb far red wavelength of light, which will also gets excited. Excited state we can symbolize as P700 star. Then the electrons will lose, then it becomes plus. Okay, now these electrons from photosystem 1 are taken by, again, a primary electron acceptor. The name of the primary electron acceptor here, what is it? It is called ferredoxin. It's taken by ferredoxin. Again, here you told the movement of electrons from photosystem 2 to pheophytin is up the hill. Here also the movement of electrons from photosystem 1 to the primary electron acceptor ferredoxin is again uphill transport. This is uphill transport. Whereas from pheophytin to plastoquinone to cytochrome B6 and to plastocyanin, it is down the hill. So this transport of electrons via electron transport system is downhill. What is meant by uphill and downhill means, basing on the redox values you're telling, when it takes electron it gets reduced, when it loses electron it gets oxidized, that way. So basing on the redox values, the movement of electrons is down the hill. But here if it is up the hill, through the electron transport system it is down the hill, and again here also it is up the hill. Right now this photosystem 1, you will need to give the electrons because it lost the electrons. It will take the electrons from plastocyanin. Means photosystem 2 electrons are coming to photosystem 1. Photosystem 2 electrons are coming to photosystem 1 via pheophytin, plastoquinone, cytochrome B6F and plastocyanin. Whereas photosystem 1 electrons are received by ferredoxin. From ferredoxin, the electrons are taken by FNR. The electrons are taken by FNR. What is FNR? FNR stands for flavin NADP reductase enzyme. It stands for flavin NADP reductase enzyme. Now this flavin NADP reductase enzyme will take the electrons. It will take the electrons. Now it is again down the hill. From the primary electron acceptor, it is again down the hill. This FNR will take the electrons and it will give the electrons to NADP reductase. No, it will give the electrons to NADP. NADP will take the electrons and it needs protons also to become NADPH2. It takes these electrons, it takes protons from the stroma and it makes NADPH2. Understood? Now, if you see here, Electrons of photosystem went to photosystem 1. Photosystem 1 electrons are finally taken by NADPH2. You need to give the electrons back to photosystem 2. That is why here we can tell there is photolysis of water, splitting of water. Splitting of water is associated with photosystem 2 or photosystem 1, they will ask you. Splitting of water is associated with photosystem 2. When you split the water molecule, you will get protons, you will get electrons, and you will get water molecule. 2 protons, 2 electrons, and half an oxygen molecule. These electrons are taken by photosystem. These electrons are taken by photosystem, and this oxygen will be evolved. This is oxygen which is evolved as a byproduct, which happens in Zscheme, which happens in non-cyclic electron transport only, right? Splitting of water happens with photosystem 2 to give the electrons to the lost photosystem 2. Now photosystem 2 electrons are taken by 1 are taken by NADPH2. Now here we told pheophytin has transported electrons to plastoquinone. Plastoquinone will transport electrons to cytochrome B6 and plastocyanin, but it has taken protons also. Those protons, what it will do? It will release into the lumen. The protons are released into the lumen. So protons are building up in the lumen. Splitting of water also happened in the lumen only, right? And plastoquinone also has released the protons in the lumen only. All these are the factors which will build a proton gradient in the lumen. Understood, children? So this is about non-cyclic electron transport chain. It is a characteristic Z scheme explained by Hill and Bendal. Take a screenshot, we will continue. Right? Now next one, we'll see cyclic photophosphorylation also. Non-cyclic was explained by Hill and Bendal, where 2 photosystems are involved. Whereas if we go with cyclic photophosphorylation, where does it occur? It occurs on the stroma thylakoid membranes. It occurs on the stroma lamellae. It occurs on the stroma thylakoid membranes. And cyclic electron transport chain starts with only one photosystem, that is photosystem 1. Cyclic photophosphorylation starts with photosystem 1. Photosystem 1 reaction center chlorophyll A is P700. P700 absorbs far red wavelength. It absorbs far red wavelength of light, and after taking that light, it gets excited. In the excited state, we have to represent it as star. Then it loses electrons. After losing the electrons, it becomes 700 plus, and these electrons are taken by primary electron acceptor. The electrons are taken by primary electron acceptor. It is up the hill. From photosystem 1, the electrons will be taken by ferredoxin, no? So these primary electron acceptor, what is it? It's ferredoxin. It gives the electrons to ferredoxin. Now, from ferredoxin, the electrons from here are going to here, and from ferredoxin, they will be taken by plastoquinone. They are taken by plastoquinone. From plastoquinone, they are taken by cytochromes. From cytochromes, they are taken by plastocyanin. From plastocyanin, the electrons will come back again. It will go to cytochrome. From cytochrome, it will go to plastocyanin. From plastocyanin, the electrons are coming back to P700 only. Now, at plastoquinone, we know it is a proton carrier also. It is a proton carrier also. Means with the electrons, it takes protons also from the stroma. Proton carrier, it takes protons from the stroma, and it helps in building up the proton gradient. This is cyclic electron transport chain. Where does cyclic electron transport chain occurs? On the stroma thylakoid membrane. Which photosystem is involved in cyclic electron transport chain? Only one photosystem is involved. That is P700. Now the movement of electrons is to the primary electron acceptor, ferredoxin, plastoquinone, cytochrome, and plastocyanin and P700. Now here if you see, is there any synthesis of NADPH2? No. Is there any synthesis of NADPH2? No, right? Because the electrons are coming back again. So they are not leaving the electrons to NADH2. There is no NADH2 made. Since the electrons are pumping back to the same photosystem, splitting of water is also not required. That's the second difference. Since splitting of water is not done with cyclic photophosphorylation, oxygen evolution also will not happen. So these are the main differences between non-cyclic and cyclic photophosphorylation. So take a screenshot. We will see the different differences between cyclic and non-cyclic photophosphorylations. Now, when we have to see the differences between cyclic and non-cyclic electron transport system. Non-cyclic electron transport system and cyclic electron transport system. Non-cyclic electron transport system was having a characteristic Z scheme, but this is having a cyclic scheme. And non-cyclic electron transport system occurs on the granathylicoid membrane. Cyclic electron transport system occurs on the stromathylicoid membrane. Non-cyclic electron transport system requires both photosystems. It requires both photosystem 2 and photosystem 1. So it requires only one photosystem, that is photosystem 1. So cyclic absorbs at, if we tell that non-cyclic it starts with photosystem 2 means at 700 nanometers, whereas if we see cyclic it uses only 680 nanometers. So non-cyclic uses two wavelengths of light. What are the two wavelengths of light? It uses 700 nanometers and it uses 680 nanometers, whereas cyclic uses only 700 nanometers of light, means it is using higher wavelength of light. It's using higher wavelength of light. Now in non-cyclic, we are getting a scheme because of the movement of electrons, right? Movement of electrons, it is uphill, down the hill, again uphill, it is again down the hill. Now here the electrons of photosystem 2 are taken by photosystem 1 and the electrons of photosystem 1 are taken by NADPH2. And electrons for photosystem 2 will come by water splitting, right? So that's what we have seen. So we have seen that photosystem 2 gives the electrons to photosystem 1. Photosystem 2 has given electrons to photosystem 1 and photosystem 1 has given electrons to NADP to make NADPH2, and this has taken electrons by splitting of water, right? So when you split, protons, oxygen and electrons will come. So photosystem 2 receives electrons from water splitting, photosystem 2 will give electrons to photosystem 1, photosystem 1 give electrons to NADPH2. Means here if you see, there is production of NADPH2. NADPH2 energy is formed. Since here it is a cyclic movement, no production of NADPH2. No production of NADPH2. And here when the plastoquinone takes the electrons, it takes protons also and it accumulates protons in the lumen. Proton gradient will be there, right? So it helps in ATP synthesis. ATP synthesis occurs here. Here also ATP synthesis occurs. ATP synthesis occurs. Synthesis of ATP is called phosphorylation. This is non-cyclic photophosphorylation. This is cyclic photophosphorylation. Synthesis of ATP is called non-cyclic photophosphorylation and this is called cyclic photophosphorylation. phosphorylation. Right, so these are some differences between cyclic and non-cyclic photophosphorylation. Non-cyclic photophosphorylation, we can stop the movement of electrons from photosystem 2 to photosystem 1 by adding an inhibitor called DCMU, dichloromethylurea. If you add dichloromethylurea, an inhibitor molecule, it stops the movement of electrons from photosystem 2 to photosystem 1. Since here such photosystems are not only one photosystem, no action of DCMU is seen here. Okay, now here NADP reductase enzyme will be there, whereas in cyclic electron transport, since there is no NADP reductase enzyme, only ATP is made, no NADPH2 is made. So NADP reductase enzyme is seen. Where is it seen? It is seen in the grana thylakoid. Whereas here we tell the stroma lamellae is devoid of, is devoid of NADP reductase enzyme. See, the stroma lamellae is not having NADP reductase enzyme also. Stroma lamellae is not having photosystem 2 also. It has only one photosystem, so that is why only cyclic electron transport system occurs. Take a screenshot, we will continue. Now, synthesis of ATP is explained by chemiosmotic hypothesis. Now let us see what is chemiosmotic hypothesis. Who explained chemiosmotic hypothesis? Arnon described it, and it works on the principle of Peter Michel chemiosmotic hypothesis. Chemiosmotic hypothesis, it explains how proton gradient is built, which is used for the synthesis of ATP, right? For that we need to see the grana. So this is one grana thylakoid membrane. The inner space is lumen. The outer part is stroma. Here is stroma and here is stroma. This is one grana thylakoid. And inside space is lumen. Now when we start Zscheme here, we will start with photosystem 2. Photosystem 2 absorbs at 680 nanometers. It gets excited and it gives electrons to pheophytin. Now this pheophytin will give electrons to plastoquinone. Photosystem 2 will give electrons to primary electron acceptor, which is pheophytin. Pheophytin will give electrons to plastoquinone. Now plastoquinone, which is located towards the outer side facing the stroma, it is not an electron carrier, it is a proton carrier. So with these electrons, it takes protons. With the electrons, it takes protons and it becomes PQH2. It becomes PQH2. The PQH2, when it is giving the electrons to cytochrome B6 and F, which is electron transport system. So PQH2, when it is giving the electrons to cytochrome B6 and F, because it is electron transport system, it will take the electrons, it will not accept the protons. Then it has to release the protons into the stroma and it becomes PQ. So PQ is responsible for accumulating the protons in the lumen. This is the first point. This is the first point. And cytochrome B6 and F will pass on the electrons to plastocyanin. Plastocyanin will pass the electrons to photosystem 1, which absorbs light at 700 nanometers. It gets excited. It gives electrons to ferredoxin. Ferredoxin will give electrons to FNR, flavin NADP reductase enzyme. Flavin NADP reductase enzyme, what it will do? It helps in synthesis of NADPH2. So FNR want to take the electrons and it want to give it to NADP plus. For NADP plus to convert to NADPH2, electrons and protons are also required. Now it takes the protons from the stroma. This is the second point, which is making the protons in the stroma less. First thing, PQ has taken protons from the stroma, made the protons in the stroma less. The second point we are telling, NADPH2 got converted, it taking electrons from photosystem 1 and it is also taking protons from the stroma, making the protons with the stroma very less. That's the second point. And photosystem 2 needs electrons because it has lost the electrons. For that, what we will do, in the lumen we will split the water molecule. You will get 2 protons, you will get 2 electrons and you will get oxygen. Oxygen will be evolved out. These electrons are taken by photosystem. These protons are also accumulated. This is the third point. So for chemiosmosis, we will tell building up of a proton gradient is required across a membrane. Means one side of the membrane proton should be more, other side of the membrane proton should be less. Now this is a granothylakoid membrane. Inside 3 points, because of 3 points protons are building more. First one, because of PQ protons are building more. Second one, because of water splitting protons are building more. Third one, because of NADPH2 protons are being consumed up. So what happens? Because of these 3 points, protons accumulate. Where do the protons accumulate? In the lumen. Protons accumulate in the lumen means protons are decreasing in the stroma. That means in the stroma protons are low, in the lumen protons are more. If the protons are more, H plus is more means pH is less. If pH is less means here pH is more here. If H is less, pH will be more here. So if the pH is less, it is creating acidic conditions in the lumen. It is creating acidic conditions in the lumen. So this builds a proton gradient. This builds a proton gradient across the grana membrane, across the membrane. Now these protons should move from high concentration. Here it is there, here it is there. They move from high concentration to low concentration. If these protons want to move from high concentration to low concentration, this is a lipid membrane. For lipid membrane, water soluble proton is not permeable. Then it has to take the help of a pump. Now that pump is made up of 2 components. It's made up of F0 and F1. These 2 together is called as ATPase. Now this ATPase helps the protons to cross the membrane. The protons from high concentration, they are coming to the low concentration. From the lumen, when they are coming to the stroma to break the gradient. So this F0 acts as a transmembrane protein. It helps the protons to move. Whereas F1, which is located towards the outer side, which is facing the stroma, has catalytic activity. It has catalytic activity. So this catalytic activity, what it will do when the protons are moving, proton motive force gets generated. With that force, ADP combines with PI to make ATP. So in this manner, ATP is synthesized in the Z scheme. This synthesis of ATP in the Z scheme is called non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, ATP is also synthesized and NADPH2 is also synthesized. Whereas in cyclic photophosphorylation, only ATP will be synthesized because electrons are coming back to the same photosystem 1. So NADP will not be synthesized. Okay. So this is chemiosmotic hypothesis, which was discussed by Arnon and which was explained by Peter Michel. It was explained by Peter Michel in the year 1961. So we can tell for chemiosmosis to occur, what are the things which are required? For chemiosmosis to occur, first one, a membrane is required. For chemiosmosis to happen, what's the first point? Membrane is required. This is the first point. Membrane is required. Then a proton pump should be there. F0, F1. A proton pump should be there. This is the second thing. A membrane is required. A proton pump should be there. A proton gradient should already be there. Here more, here less, right? In the stroma less. So a proton gradient should be there, and ATPase enzyme should be there to pump the protons. Understood, children? Take a screenshot. We will talk about non-cyclic photophosphorylation and cyclic photophosphorylation. Right. So we finished chemiosmotic hypothesis also. Now I think we can start dark reaction. We can start dark reaction, right? So we can tell in light reaction what happened. In light reaction, ATP is synthesized and NADPH2 is synthesized. Where are both the energies synthesized? In the stroma. Now in the stroma, this ATP and NADP will be used to convert. They will be used to convert carbon dioxide to carbohydrate. They will be used to convert carbon dioxide to carbohydrate. Now this light is called dark reaction. Carbon dioxide to carbohydrate conversion is called dark reaction. Where will dark reaction happen? In the stroma. Now there are two pathways to explain dark reaction. One is called as C3 cycle. Next one is called as C4 cycle. In C3 cycle, the first compound is a C3 acid. So it's called C3 cycle. Here the first compound is a C4 acid. That's why it's called C4 cycle. This was explained by Calvin. This was explained by Hatch and Slack. That's why it's called Hatch-Slack cycle. This is called Calvin cycle. Now we'll start with Calvin cycle. Take a screenshot. Before going to dark reaction, let us see the differences between photosystem 1 and photosystem 2. Then we will go to the dark reaction. Now we know that what is photosystem? It contains light harvesting complexes. It contains reaction centers. In non-cyclic, photosystem 1 and 2 both are involved. Whereas in cyclic, only one photosystem, that is 1, is involved. Let us quickly check the differences between photosystem 1 and photosystem 2. Now photosystem 1, it is located in the non-appressed region of the grana. Now what is meant by appressed and non-appressed region means, if we see this as a chloroplast and these are the grana thylakoids, now this region which is exposed, is non-expressed, and this region also you can see outside, this is non-expressed, right? It is not pressed, no, so it is non-appressed region. So we are telling photosystem 1 is present in the non-appressed region, whereas photosystem 2, we are telling it's present in the appressed region of the grana. Appressed region means this, children. This is the appressed region. This is the appressed region. This is the appressed region. Here 2 is present, and in the non-appressed region 1 is present. It is present in the non-appressed region of the grana, and photosystem 1 operates in the stroma lamella also. Photosystem 1, when it's doing cyclic photophosphorylation, where will cyclic photophosphorylation happen? Cyclic photophosphorylation happens on the stroma lamellae, right? So here cyclic photophosphorylation happens. On the grana thylakoid membrane, non-cyclic will happen. So when it is doing non-cyclic, photosystem 1 is located on the non-appressed portion. When it is doing cyclic photophosphorylation, then it is present on the stroma lamellae. Whereas photosystem 2, it comes only in non-cyclic, then it is present on the non-appressed region, it is present. This is the first difference. Second one, photosystem contains many pigments, we told. Chlorophylls, carotenoids will be there. The chlorophyll carotenoid ratio is very high in photosystem 1. Means the chlorophyll molecules are more and the carotenoid molecules are less. Then comes the ratio, chlorophyll carotenoid ratio is high here, whereas there carotenoids are more than compared to chlorophyll. That's why the numerator denominator ratio will become less. Chlorophyll ratio is less, so that's why chlorophyll carotenoid ratio will be low here. Then next one, the main pigment is chlorophyll. In that we have chlorophyll A and chlorophyll B. In photosystem 1, chlorophyll A is twice the amount of chlorophyll B. In photosystem 1, which chlorophyll is more? Chlorophyll A is more. How much it is more? It's twice the amount more than chlorophyll B. Whereas in photosystem 2, both are of equal concentrations. Now next point, photosystem 1 reaction center contains a special chlorophyll A molecule which absorbs light at red wavelength, far red wavelength, it is 700 nanometers, whereas photosystem 2 absorbs light at 680 nanometers. So photosystems are named in their order of their discoveries, not in their order of how much amount of light they are absorbing, because photosystem 2 absorbs at low wavelength, photosystem 1 is absorbing at high wavelength. Now photosystem 1 is participating in non-cyclic also, it is participating in cyclic also, whereas photosystem 2 it starts as cyclic. In photosystem, in cyclic photophosphorylation, 2 is not there. There is another difference. Now photosystem 1, we told it is there in non-cyclic also, it is there in cyclic also. In non-cyclic, photosystem 1 receives electrons from 2. In non-cyclic, it receives electrons from 2, whereas photosystem 2 will receive electrons by the photolysis of water. Photosystem 2 gets excited, it gives electrons to pheophytin. We have seen the cyclic flow. Now who will give electrons to photosystem 2? Means water will give electrons to photosystem 2. Now photosystem 1, it transfers electrons to NADP. Photosystem 1 transfers electrons to NADP. If we have to see that, if we have to see once again, photosystem 1, if it is doing cyclic electron transport, it will come back again. If it is doing non-cyclic, it starts with photosystem 2. From photosystem 2, it will go to photosystem 1. From photosystem 1, it will go to NADP reductase enzyme. So, photosystem 1 is transferring electrons to NADP and photosystem 1 is receiving electrons from photosystem 2, right? That's what I wrote here. Photosystem 1 transfers electrons to NADP when in non-cyclic. In cyclic, the photosystem 1 electrons are cycled back again. In cyclic, the electrons will return back again, whereas here if you see in non-cyclic, photosystem 2 is giving electrons to photosystem 1 and photosystem 2 will get electrons by breakage of water. 2 will give electrons to 1, it will get electrons by hydrolysis of water. So these are the differences, children, between photosystem 1 and photosystem 2. Both are present in non-cyclic, only one is present in cyclic. One is present in cyclic photophosphorylation which occurs in that stroma lamellae. 1 and 2 both are present in the granathyloid membranes where non-cyclic photophosphorylation will happen. In non-cyclic photophosphorylation, ATP and NADP both are synthesized. In cyclic photophosphorylation, since the electrons are coming back, only ATP is synthesized. So with this we are finishing light reaction. Now let's go on to the dark reaction. Now light reaction, what are the products of light reaction? Just now we discussed ATP is a product of light reaction, NADP H2 is a product of light reaction, and oxygen also is a product of light reaction. Now dark reaction is also called as biosynthetic phase. Why is it called as biosynthetic phase? Because carbohydrate is synthesized here, it is called biosynthetic phase. Why is it called as Blackman reaction? Light reactions are called Hill's reactions, dark reactions are called Blackman reactions because Blackman is a person who told that photosynthesis is biphasic and he worked mostly on the dark reactions. Now why it is called carbon dioxide fixation? The carbon dioxide is reduced to carbohydrate, that's why it's called carbon dioxide fixation. And where will dark reaction happen? It occurs in the stroma part. Light reaction happens in the grana, dark reaction occurs in the stroma. What are the products of the light reaction? ATP is one product, NADP H2 is another product, and oxygen. This oxygen diffuses out of the chloroplast and it comes in the atmosphere. That oxygen only we are inhaling. Whereas the two energy sources ATP and NADP H2 are used to make the food, are used to convert carbon dioxide to carbohydrate, right? Now Calvin at the time, during this was done at the time of Second World War, where all the scientists were too much disappointed because the radioactive discovery what they have invented is used, the radioisotopes are used in the Second World War to kill many people. So scientists were very much disappointed and they thought, like We will use radioisotopes for beneficial things only. Now then came Calvin. He used radioisotope to trace out the biosynthetic pathway. He want to see when carbon dioxides are getting joined to make a glucose, what is that path, what are the intermediate compounds he want to work out. So Calvin worked out in a green algae. He worked out in a green algae to check chlorella. He worked out in a green algae called chlorella. He used radioisotope C14 to trace out the carbon dioxide fixation pathway. He came to know that the first compound which is formed in this pathway is a 3-carbon acid. It is called as 3-phosphoglyceric acid. It is 3-phosphoglyceric acid, which is a 3-carbon acid. That is why the cycle is called a C3 cycle. It's called a C3 cycle because the first compound formed is a C3 acid. It's called Calvin cycle because Calvin worked out this cycle. So take a screenshot. We'll start with C3 cycle. Right? Now in dark reactions, there are 2 cycles to synthesize sugars. One is C3 cycle, the other one is C4 cycle. The plants where C3 cycle occurs are called as C3 plants. The plants where C4 cycle occurs are called as C4 plants. Let it be C3 cycle or C4 cycle, C3 is common. If it has to make sugar, it has to go through C3 cycle only. Now first, let us work out with C3 cycle. Now, who are those scientists who worked out means? So Melvin Calvin is the first scientist. Along with him, Benson and Bassam. They worked on a green algae called chlorella. They used radio-labeled C14 to trace out the pathway. They used radio-labeled C14 to trace out the pathway. Now C3 cycle consists of 3 steps. C3 cycle consists of 3 steps. What are the 3 steps of the C3 cycle? Now C3 cycle comes under light reaction or dark reaction? It comes under dark reaction. And where will dark reaction happen? Dark reaction occurs in the stroma. It occurs in the stroma, right? Now how many steps are there in C3 cycle? 3 steps are there in C3 cycle. 3 steps. Now step 1 is called carboxylation. Carbon dioxide is carboxylated. Now step 2 is called as reduction reaction. We will see one by one in detail. And step 3 is called as regeneration. Step 3 is called as regeneration. Now all these steps are mediated by enzymes, and all the enzymes are present in the stroma, and we tell dark reaction is sensitive to temperature because all the reactions are enzyme mediated only. Now when we have to talk about the first step, step 1, what is that called, children? Carboxylation. Now what happens here during carboxylation? So we have to take carbon dioxide molecule and we want to see who is the carbon dioxide acceptor. Who is the carbon dioxide acceptor we want to see. Now they came to know that the first stable compound which is formed is 3-PGA. So they came to know that the first stable product which is formed in the cycle is 3-phosphoglyceric acid. That is 3-phosphoglyceric acid. It is 3-PGA, which is a 3-carbon acid. Now when they have used this radio label compound, when they used this radio label C14, they came to know that the first stable compound which is formed is 3-phosphoglyceric acid. The scientists want to know that who is accepting this carbon dioxide to make a 3-carbon acid, and they were checking with a 2-carbon compound. 2-carbon compound, when it combines with 1-carbon compound, then it becomes a 3-carbon compound. That's what they were expecting. So because it came as 3-PGA, they wasted many years together in search of that 2-carbon compound. They did not find it because it's not a 2-carbon compound, it is a 5-carbon compound. Now they can ask us questions like in C3 cycle, who is the primary carbon dioxide acceptor? They can ask us in questions in C3 cycle. Now who is the carbon dioxide acceptor? If you think that carbon dioxide is making 3 PGA, now it is 3, it is 1 means it's 2 carbon compound, it is wrong. Actually the carbon dioxide acceptor in C3 cycle is a 5 carbon compound. It is a 5 carbon compound, it's a 5 carbon sugar, it is a keto sugar, it is called as RUBP. Ribulose 1,5-bisphosphate. It is ribulose 1,5-bisphosphate. Ribulose is a keto sugar. It is having 5 carbons. At first carbon it has 1 phosphate, and at the 5th carbon it has 1 phosphate. So that means we have to change the reaction as carbon dioxide is accepted by RUBP, which is a 5 carbon compound. Now in the presence of an enzyme called Rubisco, 5 plus 1, if you combine and you break it, it will get 6 carbons. 6 carbons are making 2,3 carbon acids. So this is 5 carbon compound, 1, 2, 3, 4, 5. To that, this 1 carbon compound also came. So 6. So 6 carbon compound, when you break it, you will get 1, 2, 3. 1 3 carbon compound, another 3 carbon compound. So when RUBP of 5 carbon compound combines with 1 carbon compound, then you are getting 2 molecules of, we'll get 2 molecules of 3 carbon acid. The name of the 3 carbon acid is 3-phosphoglyceric acid. It is 3-phosphoglyceric acid, and the name of the enzyme is called RUBP, carboxylase and oxygenase. This enzyme is a dual enzyme. This is a dual enzyme, means it has carboxylase activity also, it has oxygenase activity also. Means it can do carboxylation reaction, what we are doing, it can do oxygenation reaction also. When it will do carboxylase action, when it will do oxygenation action, means it will do carboxylation action when carbon dioxide concentration is high. It acts as an oxygenase when carbon dioxide concentration is low. When carbon dioxide concentration is low, that means O2 is high. When O2 is high, it functions as oxygenase. When CO2 is high, it functions as carboxylase. Now, this complete process, this complete cycle was explained by Calvin Maximum. So that's why Calvin was awarded Nobel Prize for discovery of the Calvin cycle. He got Nobel Prize in the year 1961 for explaining the complete sequence of Calvin cycle. So he worked out by using radiolabel C14 in a green algae called chlorella. He told there are 3 steps. The first step is called carboxylation. In carboxylation, what happens? Carbon dioxide combines with RUBP. It takes water molecule also. And then in the presence of an enzyme called Rubisco, which is a dual enzyme, 5 plus 1, you will get 2 molecules of 3 carbon acid, which is called as PGA. Understood, children? Take a screenshot. Right. So we have seen the first step. The end product is a 3 carbon acid, which is called PGA. When we have to draw it in the cyclic manner which they have given in NCERT, so we have to show RUBP here, which is a 5 carbon compound. It takes carbon dioxide plus water. Step 1, what is step 1? It's called carboxylation. Step 1 is called carboxylation. Now after carboxylation, we get 3 PGA. We get 3 PGA. How many molecules? 2 molecules of 3 PGA we get. And what is the enzyme? The enzyme is Rubisco. Till here we completed. Now, how many carbon dioxide should come? We know 6 CO2 plus 12 H2O. Means 6 CO2s will come. When 6 CO2s are coming, the equation becomes like 6 CO2. I'll write it on the top. We need some space. So if we take 6 CO2 plus 6 H2O plus 6 RUBP will give rise to 1 CO2 is giving rise to 2 PGAs. When you take 6, you'll get 12 molecules of 3 PGAs, right? You'll get 12 molecules of 3 PGAs, right? This is the reaction. Now, in the presence of an enzyme, Rubisco. Now, let's go to the second step. Second step is called reduction. Second step is called reduction. Now, how many phosphoglyceric acids you got for 6 cycles? So, we got 12 PGAs we got. Now, in second step, it's a series of reactions. It's not one step reduction reaction. It's a series of reaction which leads to the formation of the glucose. Reduction consists of a series of reactions which lead to the formation of glucose. Which leads to glucose formation. Right? Now here, if you start with PGA, how many PGAs are there? 12 PGAs are there. For 6 cycles, you have 12 molecules of 3-carbon acid called 3-phosphoglyceric acid. 12 molecules will take 12 ATP molecules, and it take 12 ATP molecules and 3-phosphoglyceric acid. At third place it has phosphate. ATP will give one more phosphate. It will become 12 molecules of 1,3-bis phosphoglyceric acid. Glyceric acid which is having phosphate at the third position, it takes one more phosphate from ATP and adds that phosphate to the first position. Then it becomes 1,3-biphosphoglyceric acid, and what do we get? We get 12 molecules of ADP. ATP, after donating the phosphate, it will become ADP, and the name of the enzyme will be phosphoglycerokinase. When ATP is used, it's kinase only. It is called phosphoglycerokinase is an enzyme. Now, series of reactions means one more reaction will happen. Take this 12 molecules of 1,3-bis phosphoglyceric acid. Now, it again takes 12 molecules of NADPH2. 12 molecules of 1,3-bis phosphoglyceric acid take 12 molecules of NADPH2 also, and it gets reduced. Okay, so glyceric acid is reduced to glyceraldehyde. Now, it gets reduced to 12 molecules of glyceraldehyde 3-phosphate. It gets reduced to 12 molecules of glyceraldehyde 3-phosphate. Acid got converted to aldehyde here. Okay, then next one, glyceraldehyde 3-phosphate means, that means one phosphate is released out. That phosphate comes out as 12 molecules as H3PO4, and NADP is used up here. It gets converted to 12 molecules of NADP plus, and the name of the enzyme is called as GDH, glyceraldehyde dehydrogenase is an enzyme. So if we have to see the reduction reaction once again, so in carboxylation you are having 12 molecules of phosphoglyceric acid. The 12 molecules of phosphoglyceric acid take 12 molecules of ATP. It takes one more phosphate at the first place. It becomes 12 molecules of 1,3-bis phosphoglyceric acid. Enzyme will be phosphoglycerokinase. That 12 molecules of 1,3-bis phosphoglyceric acid, now it will take 12 molecules of NADPH, and then acid gets converted to aldehyde. Triose sugar is formed. What is this glyceraldehyde 3-phosphate? It is triose sugar. How many sugars are formed? 12 molecules of trioses are formed, and the phosphate comes out as phosphoric acid, and hydrogen is used here. NADP will come. The enzyme is GDH, glyceraldehyde dehydrogenase is an enzyme. Glyceraldehyde dehydrogenase is an enzyme. So how many? For 6 cycles, so you are getting, for 6 cycles, 6 carbon dioxides. You got 12 molecules of triose phosphate, triose 3-phosphate. Triose 3-phosphate, what is the name? It is called glyceraldehyde 3-phosphate. Now, you just need to make one glucose. If you have to make one glucose, so how many trioses you need? 2 trioses if you combine. This is one triose 3-carbon sugar, another 3-carbon sugar. You will get a 6-carbon sugar. That means out of these 12 molecules, 2 triose phosphates, 2 triose phosphates are used to make glucose. 2 triose phosphates are used to make glucose. Okay. Now after you minus 2, then you are left with 10 trioses. Out of 12, give off 2, you are left with 10 trioses. This 10 trioses, glyceraldehyde 3 phosphates are excess with you. And each glyceraldehyde 3 phosphate is 3 carbon compound. How many 3 carbons are extra with you? 10. That means 10 into 30. 30 carbons are with you extra. 30 carbons are extra with you. Now, it is a cycle. What do you mean by cycle? Cycle means you have to come back to the starting compound RUBP. Regeneration of RUBP is important to keep the cycle in a flow. Regeneration of RUBP is important to keep the cycle in a flow. Now, how many RUBPs you have started or you took? 6 RUBPs you took for 6 carbon dioxide for 6 cycles, right? Each RUBP is how many carbon compound? 5 carbon compound. How many you took? 6 you took. 6 into 5, 30 carbons. You need 30 carbons to regenerate 6 molecules of RUBP back to keep the cycle in a flow. To not to interrupt it, to continue it, you need 6 molecules of RUBP. You can make it, you are having 30 carbons extra. So the third step will be regeneration. Take a screenshot, we'll go to regeneration. Right, if I have to show you that cycle, let's continue with the reduction step here in the cycle flow. This is the first step. Now in NCERT they have given the cycle for us. So 2 molecules of phosphoglyceric acid. It undergoes step 2, which is called reduction. Okay, during reduction, so for 2 molecules, it will take 2 molecules of ATP and it will take 2 molecules of NADPH2. ATP gets converted to ADP. NADPH2 gets converted to NADP plus. Now after reduction, we are getting triose 3 phosphates. We are getting triose 3 phosphates. How many triose 3 phosphates will come? 2 per cycle. We are calculating per 2, right? So 1 RUBP with 1 carbon dioxide per 1 cycle we are calculating. So 2 will come. This 2 will export to synthesize glucose or to synthesize sucrose, or they can even make starch. This is the second step, reduction. Now the third step is called regeneration. The third step is called regeneration. So I'm telling you, like RUBP regeneration is crucial. RUBP regeneration is crucial to maintain the continuity, to not interrupt the cycle. You need to regenerate, right? So if you have to regenerate 6 cycles, means 6 RUBPs, means each RUBP is 5 carbon compound, means 6 5s are 30, which are there with us. Now in regeneration, what happens means, in regeneration, let's go to the third step. It is step 3, regeneration. Now in regeneration, for each RUBP, it takes 1 ATP, it gives out 1 ADP. For 1 cycle, 1 ATP and 1 ADP. If you see the energy yields per cycle, now in step 2, it has taken 2 ATP and it has taken 2 NADPH2. And in step 3, it has taken 1 ATP. Altogether, you can tell 3 ATPs plus 2 NADPH2 per cycle. How many cycles should operate? 6 cycles, right? Because 6 carbon dioxides. 6 cycles, 6 carbon dioxides, that means 6 into 3. 6 3s are 18 ATPs and 6 2s are 12 NADPH2s are required. Means 6 extra ATPs are required than NADH2. That is why to compensate this extra number of ATP, cyclic photophosphorylation will happen. Cyclic photophosphorylation will synthesize only ATP. Non-cyclic will synthesize ATP and NADP. To compensate this more ATP, cyclic photophosphorylation will occur. Cyclic photophosphorylation will occur. Understood, children? Now at the end they told in C3 cycle what are you taking in and what is coming out. Just now we calculated 6 CO2s we take, and what will come out is 1 glucose. What will come out is 1 glucose. There's a table in NCERT. How many ATPs are you taking? So 6 3s are, in the second step 2 ATP, in the third step 1 ATP. So altogether we take 18 ATPs, gets converted to 18 ADPs. And how many NADPH2s? Only in the second step 2 NADH2s. So 2 into 6, 12. So altogether 12 NADPH2s are used and they get converted to 12 NADPs. So this we will take and this we will get. So this is a energy consumption of C3 cycle. This is energy consumption of C3 cycle. Now C3 cycle, what is C3 cycle? C3 cycle is called Calvin cycle. It is called phosphoglyceric acid cycle. The first stable compound is phosphoglyceric acid. Right? Who discovered it? Calvin discovered it in the year 1961. So I think you understood. They will ask you how many ATPs are required in reduction per cycle? 2 ATPs. How many ATPs are required in regeneration per cycle? 1 ATP. How many ATPs are required per cycle, altogether per cycle? 3 ATPs. Like that, on this concept they can ask you a number of questions. So take a screenshot, then we will start with C4 cycle. Now let us talk about another pathway which is called a C4 pathway, which also comes under dark reaction. C4 pathway. So earlier we have seen C3 pathway, which was worked by Calvin, for which he got Nobel Prize in the year 1961. There's another pathway to make sugars in some plants which is called as C4 pathway. It has many names, like it is called as beta carboxylation pathway. It is called beta carboxylation pathway because 2 times carboxylations will happen. We are calling it as beta carboxylation pathway. Now, the major part of the cycle was worked by 2 scientists called Hatch and Slack. So it's called Hatch-Slack pathway also. The other name is, it is called Hatch-Slack pathway. But initial investigations are done by another scientist called Kortschak. So since this scientist is also involved, so we can also call it as HSK pathway. So after the names of these scientists, Hatch, Slack and Kortschak, we can also call it as HSK pathway. Now when we have to talk about the historical concepts about C4 pathway, we have to tell that Kortschak, Hart and Burr, they found that in some plants, the first product is not a C3 acid, is not a 3-phosphoglyceric acid, it's something extra, it's something a different one which is having 4 carbons in it. So then, since the first compound formed in these plants is a 4-carbon acid, it is called as C4 pathway. Who made the discovery? These 3 scientists. First scientist is Kortschak, Hart and Burr. Now these 3 people reported that in sugarcane plants, the first compound or the first product is not a 3-carbon compound. What did they observe? They observed that in particularly sugarcane plant, the first stable product which is formed in the dark reaction is not a 3-carbon compound. Instead, they told it's a 4-carbon compound. Instead, it is a 4-carbon compound, they told. Right? And they told the 4-carbon compound can be oxaloacetic acid, it can be oxaloacetic acid, it can be malic acid also. Now if you see, oxaloacetic acid also has 4 carbons, malic acid also has 4 carbons. So in that, 2 carbons will be on the backbone and 2 carbons are acidic groups. So we can also call it as dicarboxylic acid. Since 2 acid groups are there, we can also call it as dicarboxylic acid. So they found that it is not a C3 carbon acid, it's a 4 carbon acid. It can either be oxaloacetic acid or malic acid. So these two are 4 carbon compounds having carboxylic acids. They have 2 acids in them. Okay, now later Hatch and Slack proved that yes, it is correct only. These people told it, and who proved it? Hatch and Slack confirmed, right? They confirmed it is oxaloacetic acid only. So that is why they called the cycle as C4 cycle. They call the cycle as C4 cycle. Now if we see, like, the plants which are exhibiting C3 cycle are called C3 plants, and the plants which are showing C4 cycle are called as C4 plants. And we tell C4 plants are more biologically evolved than C3 plants. So I'm giving a statement that C4 plants are more biologically evolved than C3 plants. Means I need to justify the statement. How are C4 plants more evolved than C3 plants? Means if we see, C4 plants can tolerate high temperature. C4 plants can tolerate high temperatures. Means C4 plants can work in the tropical and subtropical regions. C4 plants can work in tropical and subtropical regions where the temperature is 30 to 45 degrees centigrade. Where the optimal temperature, how much it is? It is more. It is 30 to 45 degrees temperature. Whereas C3 plants, if you take, they can tolerate only 20 to 25 degrees temperature. Whereas C4 plants, they are tolerating higher temperature. This is one point. And next point, we can tell that C4 plants are drought tolerant or drought resistant. Means they can tolerate water stress. They can tolerate water stress even if you are not giving water, so they can tolerate it. Means they are tolerant to water stress. They are drought tolerant plants. They are tolerant to saline conditions also. C4 plants are tolerant to saline conditions also. Salty conditions also they can grow. This is another advantage we are telling. And then C4 plants, like they show a particular anatomy that's called as Kranz anatomy. C4 plants exhibit Kranz anatomy. Now the meaning of Kranz in German is Reth. Reth means ring. Usually we use like 4 floral whorls, we use it for flower. 4 rings are there. So here in the plant we are using a word called reth. We are using a word called ring. Okay, in C4 plants, if we see the leaf, in C4 plant, if we see the leaf, if this is the upper epidermis and this is a lower epidermis. So these are the upper epidermal cells. I'm showing a stomata here, and these are the lower epidermal cells. And in between these two are parenchymatous mesophyll cells. These are parenchymatous mesophyll cells, right? They are arranged like pillars, so they're called palisade parenchyma. And here it has vascular bundle. It has vascular bundle. In the vascular bundle, this much patch is called as a phloem patch, and here are xylem. Protoxylem and metaxylem. Now in C4 plants, if we cut the C4 plant, sugarcane plant or amaranthus plant or pearl millet, all these are examples of C4 plants. If you cut the leaf for C4 plant, we can see that the vascular bundles are surrounded by one more ring. That is why it's called Kranz anatomy. Reth, there is a ring. Ringed vascular bundles are present. Means this vascular bundle cells are covered by ring-like cells which are called bundle sheath cells. Like a bundle, they are covering. And one layer of bundle sheaths are, or many layers of bundle sheaths are. The vascular bundles are surrounded by many layers of bundle sheath cells. And what is the speciality of these bundle sheath cells? These bundle sheath cells have thick walls. I draw the bundle sheath. I draw 2 layers. So that's called bundle sheath. The bundle sheath cells have thick walls. They are having many layers and the cells are compactly placed. The cells are compactly spaced. There are no spaces. You can see here, their compactly placed spaces are not there, and they have Casparian thickenings also. These bundle sheath cells, they will have suberin depositions. They will have Casparian thickenings also. This Casparian thickening, the presence of this Casparian thickenings on the radial walls and the tangential walls will not allow the gases to permeate inside. So these Casparian thickenings are giving impermeabilis nature for the gases. So no gas exchange occurs. No gas exchange occurs. So such type of anatomy is found only in the C4 plants. What is that anatomy called? Kranz anatomy. What do you mean by Kranz anatomy? The vascular bundles are surrounded by many layers of bundle sheath, and these bundle sheath cells are thick cells. They are tightly packed without any spaces. They are having Casparian thickenings also, which makes it impermeable for gases exchange. It will not allow any gas to enter inside. And not only that, these, if we talk about chloroplast, usually the chloroplast will be there in the mesophyll cells. I draw the mesophyll cells. So these are palisade mesophyll cells and these are spongy parenchymatous mesophyll cells. Usually the chloroplast in C3 plants will be there in these mesophyll cells. The dots which I am keeping represents the mesophyll cells. So usually the chloroplasts are present in the mesophyll cells if it is a C3 plant. But now we are drawing C4 plant. In C4 plant, now what happens? The chloroplast along with the mesophyll cells, they are also present in the bundle sheath cells. Now these are bundle sheath cells. In the bundle sheath cells also I should show chloroplast. Am I doing that? I'm showing chloroplast here, and if you notice, I'm keeping many dots inside the bundle cells. Means I want to tell you that the bundle sheath cells are having more number of chloroplasts, large number of chloroplasts than the mesophyll cells. The number of dots here are limited, but here I'm packing it too much. So that means the bundle sheath cells of the C4 plant surrounding the vascular bundle will have large number of chloroplasts. Understood? So this is about Kranz anatomy. Take a screenshot. We'll continue. Right now, since these many advantages, these many special features are there, we are telling C4 plants are more biologically evolved than C3 plants. You'll understand slowly when we go into the topic. What are the other advantages? The one advantage C4 plant is having is Kranz anatomy. The other advantage, I'll tell, chloroplast dimorphism. What do you mean by chloroplast dimorphism in C4 plants? Means, come on, in C3 plants, chloroplast is present only in these mesophyll cells. But in C4 plants, chloroplast is there in the mesophyll cells also, chloroplast is there in the bundle sheath cells also. Two types of chloroplasts are present. So that's why we call it as chloroplast dimorphism. The first point we told, C4 plants will exhibit Kranz anatomy. And the second point we are telling, C4 plants show chloroplast dimorphism. C4 plants show chloroplast dimorphism. What do you mean by chloroplast dimorphism? Means there are two types of chloroplast. One chloroplast is there in the mesophyll cell. The other chloroplast is there in the bundle sheath cell. The chloroplast which is there, one chloroplast is there in the mesophyll cell, another chloroplast is there in the bundle sheath cell. So the chloroplast which is there in the mesophyll cell is called as granule chloroplast. Whereas the chloroplast which is there in the bundle sheath cell is called agranule chloroplast. Now, what do you mean by granule chloroplast? Granule chloroplast means if we draw the chloroplast, outer membrane and the inner membrane, and these are the membrane thylakoid sac-like structures which are called as granum. Many granums is called as grana, and they are interconnected by stroma lamellae, right? They are interconnected by these structures called stroma lamellae. So if it is having grana thylakoid and if it is having stroma thylakoid, it's called granule chloroplast. Whereas in the bundle sheath cells, I'm telling the chloroplasts are agranule chloroplast. Agranule chloroplast means chloroplast will be there, inner membrane and outer membrane, but since we are telling they are agranule, this granum is absent. Granum is absent means then what is there? Only stroma lamellae are there. So we can tell the bundle sheath chloroplast will have only stroma lamellae. So I am drawing stroma lamellae, see. They are having only stroma lamellae. Grana are absent here. Since two types of chloroplasts are there, we call it as chloroplast dimorphism. Chloroplast dimorphism is exhibited by C4 plants. Now, how many plants are there which come under this category of C4 plants? Means 1500 species of angiosperms are identified. 1500 species of angiosperms have been identified which are exhibiting C4 pathway, and they come under 19 families. They are grouped under 19 families. Altogether there are 1500 species. If you have to list down some examples of C4 plants, we have already told sugarcane. Another example we can give maize. Another example we can give sorghum. The examples of C4 plants are, so they have connected exponential sugarcane. We can take sugarcane. Sorghum we can take, and then maize we can take. Pearl millet, amaranthus, right? So Cynopodium. These are some examples of C4 plants, right? We told they are 1500 in number. They belong to 19 families, and these are some examples of C4 plants. Take a screenshot. We'll talk about C4 cycle. Right? So we finished a peculiarity. Now let us get into the cycle. How many steps are there in C4 cycle and how does it occur? If there are two types of chloroplasts, then some reactions of C4 cycle will happen in the granule chloroplast and some reactions will happen in the agranule chloroplast. So that's why in NCERT for C4 cycle they showed us two chloroplasts. The chloroplast in the mesophyll cell also, the chloroplast in the bundle sheath cell also. Let us draw. First we'll draw the mesophyll cell because near to the epidermis mesophyll chloroplast will be there. So this is the mesophyll cell chloroplast. Okay, and then they are interconnected by plasmodesmatal connections. And then next we are going to draw bundle sheath cell chloroplast. Right? So now this is a plasmodesmatal connection which is connecting mesophyll cell chloroplast to bundle sheath cell chloroplast. Now what is this? And what is this? Bundle sheath cell. And what is this connection called? Plasmodesmatal connection. This is plasmodesmatal connection. Now, what are the steps of C4 cycle? Means there are 4 steps. The first step is called carboxylation. During carboxylation, now the atmospheric carbon dioxide comes inside the mesophyll cell as bicarbonate ion. So the atmospheric carbon dioxide is taken inside. When the stomata opens, carbon dioxide comes into the mesophyll cell's chloroplast as bicarbonate ion. In C3 cycle we told the primary carbon dioxide accepted in C3 cycle is a 5-carbon compound called Rubisco. Now here who will accept this carbon dioxide? Means the carbon dioxide is accepted by a 3-carbon compound called phosphoenol pyruvate. So PEP stands for phosphoenol pyruvate. Phosphoenol pyruvate. How many carbon compound is this? It is a 3-carbon compound. 3-carbon compound, when it combines with 1-carbon compound, then what happens? 4-carbon compound is formed. What is this reaction called? Fixation, carboxylation or fixation. In NCERT, they showed us it is fixation. We can also call it as carboxylation. And now during fixation, a C4 acid will be formed. 3-carbon acid with 1 carbon, 4 carbons. A C4 acid will be formed. And this enzyme here is called pepcase. Phosphoenol pyruvate carboxylase. What's the full name of pepcase? The enzyme which will mediate the first reaction is called phosphoenol pyruvate carboxylase. Now, this first reaction, where does it occur? It occurs in the cytoplasm of the mesophyll cell only. The first reaction occurs in the cytoplasm of the mesophyll cells only. Now, the acid which is formed here is called as oxaloacetic acid. The acid which is formed as the end product of the first reaction is called oxaloacetic acid. Since it's a 4-carbon acid, we are calling the cycle as C4 cycle. Now, this oxaloacetic acid from the cytoplasm of the mesophyll cells, it enters into the chloroplast. Now, it enters into the chloroplast, and there itself this oxaloacetic acid gets converted to malic acid. There itself the oxaloacetic acid gets converted to malic acid. The enzyme is called malic enzyme. In the first reaction, the enzyme is called pepcase. Now here the enzyme is called as malic enzyme, which converts a 4-carbon acid into another 4-carbon acid. Oxaloacetic acid gets converted to malic acid. Where is this reaction happening? Inside the chloroplast of the mesophyll cells. Where did the first reaction happen? In the mesophyll cell cytoplasm only, not inside the chloroplast. Now this malic acid which is formed will get transported into the bundle sheath cell. The 4-carbon acid from the mesophyll cell through the plasmaresthematic connections, it is entering into the bundle sheath cell. After entering into the bundle sheath cell, now what happens? This undergoes decarboxylation. Now this 4-carbon acid which entered here, it undergoes decarboxylation. Means from a 4-carbon acid, when you remove carbon dioxide, from a 4-carbon acid, when you remove a carbon dioxide, then it becomes a C3 acid. So after decarboxylation, we'll get C3 acid. This C3 acid is called pyruvic acid. What is the C3 acid called? The C3 acid is called pyruvic acid. Understood, children? Now, this enzyme which will do this reaction is called, what is this reaction called? Oxidative decarboxylation. The first reaction, what is the first reaction? First reaction is bicarbonate combines with phosphoenol pyruvate. Then it gets converted to a 4-carbon acid called oxaloacetic acid. Now, this is mediated by an enzyme called pepcase, right? Now the second reaction is conversion of this oxaloacetic acid to malic acid, right? So what happens means the oxaloacetic acid takes up NADPH2 and gets converted to malic acid, and NADH2 will be formed. This is the second reaction. What is this reaction called? This reaction is conversion of one 4-carbon acid to another acid. This is mediated by an enzyme called malate dehydrogenase. It's not malic enzyme. This is mediated by an enzyme called MDH, malate dehydrogenase. Because hydrogen is involved here, we are calling it as malate dehydrogenase. Now the third step is malic acid is undergoing oxidative decarboxylation. Malic acid is undergoing oxidative decarboxylation, right? So take the malic acid, which is a 4-carbon acid, and take we have to take NADP. Now it gets converted to a 3-carbon acid by releasing carbon dioxide. Carbon dioxide will come and pyruvic acid will come and hydrogens will go off. Again we are generating NADPH2. The NADPH2 which is consumed here is synthesized back here. The NADPH2 which is consumed here is synthesized back here, and this enzyme is called as malic enzyme. Oxidative decarboxylation is conducted by malic enzyme. Now you got pyruvic acid. So here is a pyruvic acid which is a C3 acid. This pyruvic acid is transported back. It is transported back from the bundle sheath cell into the mesophyll cell. The pyruvic acid came off from the mesophyll cell. C3 acid came off from the mesophyll cell into the from the bundle sheath cell into the mesophyll cell. Now this pyruvic acid has to do the fourth reaction which is called as regeneration. This is called as regeneration. So in order to continue the cycle, we need to convert the pyruvic acid into phosphoenol pyruvate. The difference between pyruvate and phosphoenol pyruvate is having phosphate. So if you give ATP, pyruvic acid gets converted to phosphoenol pyruvate, right? So now in this reaction, which is called regeneration of phosphoenol pyruvate, it takes ATP. Each pyruvic acid actually it requires 1 ATP, but it demands 2 ATPs and it converts to 2 ADPs. So that's why the enzyme is called as pyruvate dikinase. Even though it requires only 1 ATP, it demands 2 ATPs. So that's why the enzyme is called pyruvate dikinase. Regeneration. In regeneration, each pyruvic acid molecules take 2 ATPs and then converts the pyruvic acid into phosphoenol pyruvate and 2 ADPs. The name of the enzyme is called pyruvate dikinase. Pyruvate dikinase. Understood? So we are doing for 1 carbon dioxide. It will be done for 6 carbon dioxides, right? 6 CO2 plus 12 H2O. 6 times when you are doing, then what happens? It will become 12. Then it will become 12. Now this is C4 cycle. Excuse me. C4 cycle. First one is PEPKase action, second one is MDH action, third one is oxidative decarboxylation, and the fourth one is regeneration. Now what happens to the carbon dioxide which is there in the bundle sheath cells? The carbon dioxide which is released in the bundle sheath cells will go to C3 cycle. It will go to C3 cycle to synthesize sugar. Now make a point here, let it be C3 plant or a C4 plant, if it has to synthesize sugar, it has to enter C3 cycle only. That means in C4 plants also, for synthesizing sugar, C3 pathway is only occurring. But in C3 plants, C3 pathway occurs in the mesophyll cell. In C4 plants, C3 cycle occurs in the bundle sheath cells. Okay. So that means there are 2 decarboxylations I told. There are 2 carboxylations I told. One carboxylation is here, when it comes in and it is fixed. The second carboxylation here. Here the carbon dioxide is accepted by PEP. Here the carbon dioxide is accepted by RUBP. C3 cycle we know, right? Carbon dioxide is accepted by RUBP to make 3PGA. That is the flow. Take a screenshot. We'll continue. Right? Now, when we talk about the energetics of C3 cycle, we told in C3 cycle, in C3 cycle, how many in and how many out? In C3 cycle, 6 carbon dioxides will go inside and 1 glucose will come outside. And C3 cycle, it takes 18 ATP, it uses 18 ATP, converts it to 18 ADP. And it takes 12 NADPH2 and convert them into 12 NADPs. So now if we have to, this is for C3 cycle. Now if we have to talk for C4 cycle, in C4 cycle also C3 cycle is operating. That means these things are there. Along with that, is there any additional requirement of energy? Yes. Where did we see that energy? Here ATPs are required. For 1 pyruvic acid, 2 ATPs. But for 1 pyruvic acid, for 1 carbon dioxide, how many carbon dioxides will enter? 6 carbon dioxides will enter. No. For 6 carbon dioxides to make 1 glucose, C6H12O6. So it requires 12 then. For 1, 2, that means for 6, 6 2s are 12. So that means it requires 12 additional ATP. 12 additional ATP with this 18 ATP of C3 cycle. So 12 plus 18 all together, if you take C4 cycle plants will take 30 ATPs, but NADPH2s are the same, because in the second step NADPH2 is used, in the third step NADPH2 is made back again, so it becomes 0. So the NADP productions are 12 NADPH2s only. So this is the energy requirement for C4 cycle. This is the energy requirement for C4 cycle. Hope you understood about C4 cycle. Take a screenshot. Then we will talk about the comparisons of C3 and C4 plants, which is given in NCERT in the form of a table. Now let us see the differences between C3 and C4 plants, which is given in NCERT table, right? So I have taken all the points. We'll fill up like what characteristics are applicable for C3 plants and what characters are applicable for C4 plants. This table is there in NCERT textbook also. Now we have seen C3 pathway, we have seen C4 pathway. Why it is called C3 pathway? The first product which is formed in these plants is a 3 carbon acid, so that's why it's called C3 pathway. Why this is called C4 pathway? The first product which is formed is a 4 carbon compound, that is why it's called C4 pathway. So the first point, cell type in which Calvin cycle occur. So we told let it be C3 plant or C4 plant when it is doing photosynthesis or when it's doing sugar synthesis, then it has to follow C3 pathway only. C3 pathway means Calvin pathway only. Now the cell type in which Calvin cycle occurs in C3 plant, Calvin cycle occurs in mesophyll cells, and in C4 plants Calvin cycle occurs in the bundle sheath cells, right? It occurs in the bundle sheath cells. Now in C3 plant, the sugar synthesis occurs in which cell? Mesophyll cell. Whereas in C4 plant, it occurs in the bundle sheath cell. Next one, cell type in which the initial carboxylation reaction occurs. Now in C3 plant, the first carboxylation reaction occurs in the mesophyll cell only, and in C4 plant also, the first step, carbon dioxide, bicarbonate ion combines with PEP to make a 4-carbon acid, oxaloacetic acid, is in the mesophyll cell only. So we can write here, the first carboxylation reaction occurs in which cell means here it is in the mesophyll cell for C3 plant, and in C4 plant also it is mesophyll cell only. In C4 plant also it is mesophyll cell only. Next one, how many types of cells does the leaf has to fix carbon dioxide? In C3 plants there is only one cell where the cycle will happen, it is a mesophyll cell. But in C4 plants 2 types of cells are there. One is mesophyll cell, another one is bundle sheath cell, which is enclosing the vascular bundle. So how many cell types if they ask means we have to give only one cell type. What is that one cell type? Mesophyll cell. Whereas here I am telling there are 2 cell types. What are the 2 cell types? So one cell is a mesophyll cell and the other one is a bundle sheath cell. Right? Now next one, which is a primary carbon dioxide acceptor? In C3 plants the carbon dioxide is accepted by a 5-carbon compound called RUBP. So which is a primary carbon dioxide acceptor here? In C3 plants it is ribulose 1,5-bisphosphate. Okay, in C4 plants the carbon dioxide is first accepted by PEP, phosphoenol pyruvate. So here it is phosphoenol pyruvate. Then number of carbons in the primary carbon dioxide acceptor. In C3 plants the primary carbon dioxide acceptor is RUBP. I told it is a 5-carbon compound. Number of carbons in RUBP, 5 carbon compounds. Number of carbons in PEP, 3-carbon compound it is. Now which is the primary carbon dioxide fixation product? Now in C3 plants RUBP combines with carbon dioxide to make a 3-carbon acid which is called 3-phosphoglyceric acid. So which is the primary carbon dioxide fixation product here? The primary carbon dioxide fixation product is 3-phosphoglyceric acid. It is 3-phosphoglyceric acid here, which is a 3-carbon acid, and that is why it is called C3 cycle. Whereas in C4 plants the primary carbon dioxide fixation product is a 4-carbon acid which is called oxaloacetic acid or malic acid. So here it is oxaloacetic acid or malic acid, which is a 4-carbon acid. Right? Next one, does the plant has rubisco? Rubisco is the most abundant protein of the plant kingdom we can tell, or the biosphere we can tell, or in the world we can tell. Does the plant has rubisco? In C3 plant RUBP combines with carbon dioxide. In the presence of rubisco only it will form PGA. So rubisco is present in this plant also. Rubisco has to be present in this plant also, because photosynthesis or the sugar synthesis happens with rubisco only. Does the plant has rubisco? Yes, it has rubisco. Here also it has rubisco. Next one, does the plant has PEPKs? In C3 plants PEPKs will not be there. In C4 plants PEPKs will be there. So does the plant has PEPKs? In C3 plants no. In C4 plants yes. Which cells in the plant has rubisco? Now rubisco is present in the mesophyll cell in the C3 plant, and the Calvin cycle operates in the bundle sheath cell there. So rubisco will be present in the bundle sheath here. Which cell in the plant has rubisco means here it is a mesophyll cell. Whereas in C4 plant it is bundle sheath cell. In C4 plant it is bundle sheath cell. Now carbon dioxide fixation rate at high light intensity. Carbon dioxide fixation rate at high light intensity. Now C3 plants it will be moderate. C4 plants tolerate higher temperatures, 30 to 45 degrees temperature. So carbon dioxide fixation rate at high light intensity for C3 plant it is moderate. For C4 plant, since they can tolerate high temperature, carbon dioxide fixation rate at high light intensity will be high. Now, whether photorespiration is present at low light intensity. So, after finishing this, we'll talk about photorespiration, which is a waste process. Why I'm telling you it's a waste process means photosynthesis stops and respiration starts. When does it happen? Means when the carbon dioxide concentration is low, when the oxygen concentration is high, right? So, and C3 plants are more susceptible for photorespiration. C4 plants they don't show photorespiration. That's why we tell C4 plants are more evolved than C3 plants. Whether photorespiration is present at low light intensities. At low light intensity, neither this nor this will do photorespiration. It's negligible, you can tell. And here also at low light intensities, both will not do photorespiration. Whether photorespiration is present at high light intensities, yes, C3 plants will do photorespiration at high light intensities, but C4 plants negligible, they don't do photorespiration. They don't do photorespiration. Now, whether photorespiration occurs at low carbon dioxide levels, yes, I told you, photorespiration happens when the carbon dioxide levels are low and when the oxygen levels are high. So, whether photorespiration occurs at low carbon dioxide, so we need to tell yes. Only C3 plants will do photorespiration, no. C4 plants will not do photorespiration, so no. Now, whether photorespiration occurs at high carbon dioxide, when the carbon dioxide concentration is high, then it can convert that carbon dioxide into carbohydrate only. So, when the carbon dioxide concentration is high, this will also not do photorespiration. This anyway, it will not do photorespiration. So, this will not do, this will also not do. Optimal temperature. I told the optimal temperature for C3 plants is 20 to 25 and for C4 plants is 30 to 45. So the temperature optimum for C3 plant is 20 to 25 degrees centigrade and for C4 plants it is 30 to 45 degrees centigrade. Now carbon dioxide saturation point. C3 plants will saturate at higher levels and C4 plants saturates at lower levels. So carbon dioxide saturation point is high in C3 plant and carbon dioxide saturation point is low in C4 plant. Now in C3 plant, the carbon dioxide compensation point at which level it saturates means high means it saturates at 460 microliters per liter. Whereas C4 plant low means it is at 350 microliters per liter, right? It is 360 here and 450 here. Carbon dioxide saturation point in C3 plants is high, means how much high? It's 450 microliters per liter. In C4 plants it is low, means how much low? 360 microliters per liter. Now number of ATPs and NADPs required for one glucose molecule. Now we calculated for one glucose molecule, if it is doing C3 cycle, 18 ATPs and 12 NADPs are required. Number of ATPs and NADPs required to make one glucose is 18 ATPs and 12 NADPH2s are required. Whereas in C4 plants, 12 extra ATPs will be required for regeneration. So 18 plus 12, 30 children. So here it is 30 ATPs, but NADs are the same only. 12 NADPH2s only. Now the last one they're asking are the examples for C3 plants. Majority of the plants are C3 plants only, right? So majority of the plants we can tell. C4 plants will list down some examples. For C4 plants we'll list down some examples. I'll write down the examples of C4 plants here. So we have already seen examples are amaranthus is one example. Sugarcane is another example. Sorghum is another example. Pearl millet is another example. Maize, Chenopodium. These are some examples of C4 plants. Children, these are the differences between C3 and C4 plants. Take a screenshot, then we will talk about the next topic, which is photorespiration. We'll discuss about the next topic, which is photorespiration. Now, we are discussing that photorespiration is seen in C3 plants, is not seen in C4 plants. Now, let's try to understand what is photorespiration. So, C3 cycle, the first compound is C3 compound. C4 cycle, the first compound is C4 compound. In photorespiration, the compound which is formed is 2-carbon compound. That's why photorespiration is also called C2 cycle, right? Now, who observed this? Means Warburg observed a phenomenon. That's why photorespiration, we can also call it as Warburg effect. What did he observe? He observed that in algae, when the carbon dioxide concentrations are low, obviously it means that oxygen concentrations are high. He told that green algae, when the carbon dioxide concentration is low and when the oxygen concentration is high, photosynthesis is inhibited. In the presence of oxygen, photosynthesis will not happen. That's what is his conclusion, which is called as Warburg effect. In 1920, Otto Warburg observed that in the presence of oxygen, in the presence of more oxygen, photosynthesis will stop and respiration will start. It inhibits photosynthesis. This phenomenon is called Warburg effect. Understood? And who worked out the process of photorespiration means Decker and Theo. In which plant? In tobacco plant. So, the process of photorespiration was demonstrated in tobacco plants by Decker and Theo. Who observed that photorespiration is there? Warburg observed. Who demonstrated in tobacco plant? Decker and Theo demonstrated it, right? Which plants will do photorespiration? C3 plants will do photorespiration. So, we'll see why C3 plants are susceptible to photorespiration. So, C3 plants are sensitive to this photorespiration cycle, while C4 plants are not affected, right? This process occurs in 3 organelles. Photorespiration happens in 3 organelles. I use 3 colors here. So, it happens in chloroplast, it happens in peroxisomes, it happens in mitochondria. So, photorespiration occurs in 3 organelles. The first 2 reactions in the chloroplast, next 2 reactions in the peroxisomes, next reaction in the mitochondria, like that. It occurs in 3 organelles. Now, we know the most abundant protein in the whole biosphere in the world is rubisco, right? Rubisco is an enzyme. RUBP, carboxylase and oxygenase. So, one enzyme is having 2 effects. Carboxylation, it can do. Oxygenation also it can do. That's why it's called carboxylase and oxygenase. So, rubisco is the most abundant protein in the world, in the biosphere. Rubisco has dual enzyme. It is a dual enzyme. It can function as carboxylase, it can function as oxygenase. When the carbon dioxide concentration is high, it will do carboxylation activity. When the oxygen concentration is high, it will do oxygenase activity. So, when carbon dioxide and oxygen are equal, then it has more affinity for CO2 only. When carbon dioxide is high, it will do photosynthesis. When oxygen concentration is high, it will do photorespiration. But if both are at equal levels, then it has more affinity for CO2 only. Then it will do photosynthesis only. Okay. So, at equal concentrations of CO2 and O2, rubisco has more attraction for CO2. If the rubisco is taking CO2, fixation happens. Then when fixation happens, C3 acid will come, PGA will come. At high levels of O2, CO2 is lesser, means O2 is high. When the oxygen concentration is high, then it will function as oxygenase. Then those extra molecules of oxygen will go and bind to the rubisco's active site first and they will do respiration. So, at high levels of oxygen, competitive binding, oxygen is showing more competition and it's going and bind to rubisco and it starts photorespiration. It's doing respiration by stopping photosynthesis. So, that's why we call it as a photorespiration. Now, in C3 plants only, the mesophyll cells will be exposed to the atmosphere and in the atmosphere 21% oxygen is there. So, rubisco will become sensitive in the C3 plants because where is C3 cycle happening? In the mesophyll cells. Where are mesophyll cells? Adjacent to the atmosphere. Okay. So, in C3 plants, some oxygen binds to the rubisco. So, that's why C3 plants will give less yield. Whereas in C4 plants, we told chloroplast dimorphism. 2 types of chloroplasts are there. One chloroplast in the mesophyll cells, another chloroplast in the bundle sheath cells which are surrounding the vascular bundles which are inside. So, in the bundle sheath cells, the rubisco will hide. Inside the bundle sheath cells, the rubisco will hide. It's not in the mesophyll cells, right? So, and again, if we talk about the bundle sheets, they have thick walls, they are tightly packed, they are having Casparian thickenings, they are impervious to gases. Means oxygen, can it come into the bundle sheath cells? No. So that means the rubisco in C4 plants, it is hiding inside where oxygen cannot come. Then there is no question of photorespiration. It can do only photosynthesis. That's why the yield will be more. So in photorespiration, there is neither synthesis of sugar. We call photorespiration as a waste process. Why are we calling it as a waste process? Synthesis of sugar happening? No. Or at least ATP is getting synthesized, energy is getting synthesized? No. Instead, ATP is used, wastage of energy, and carbon dioxide is released. Here you can see carbon dioxide is released. See, carbon dioxide is toxic. So two bad things it is doing. It's increasing the carbon dioxide concentration and it is consuming ATP. Two good things it is not doing. It's not making food nor it is making ATP. So that's why we call it as a waste process. In C4 plants, what happens? Photorespiration is not there. In C4 plants, they have a mechanism to increase the carbon dioxide concentration at the rubisco. 2 times they will do decarboxylation, right? So the carbon dioxide concentration at the bundle sheath cells where rubisco is there will be more. And the bundle sheath cells in the C4 plants are having Casparian thickenings. They are impervious for gases exchange. So oxygen cannot go inside. They do not show photorespiration. So children, this is the information about photorespiration. If we have to talk about the cycle, here I have taken the cycle. So let us discuss. Now we told photorespiration will happen when the CO2 concentration is low, when the oxygen concentration is high. See, I wrote here when oxygen is high. When the oxygen is high, then RUBP, a 5-carbon compound. A 5-carbon compound in the presence of oxygen, it will split into a 3-carbon compound and a 2-carbon compound. The 3-carbon compound, we know it is PGA only. So RUBP in the presence of oxygen, it will split into a 3-carbon compound. Then the leftover 2-carbon compound is called phosphoglycolate. So RUBP, which is a 5-carbon compound, when it takes oxygen, it is split into 2-carbon compound called glycolate, 3-carbon compound called phosphoglyceric acid. Okay, this phosphoglyceric acid will do C3 cycle, will do Calvin cycle. But what happens to the 2-carbon glycolate? This question has been asked in NEET 2020 examination. They asked when RUBP binds with oxygen. When RUBP binds with oxygen, what are the products we are getting? RUBP is a 5-carbon compound. When it binds with oxygen, then the 5-carbon compound is broken down into a 3-carbon compound called PGA, and the 2-carbon compound is phosphoglycolate. It is phosphoglycolate. Then this phosphoglycolate loses phosphate and it becomes glycolate. So this is the first reaction. This is the second reaction which is happening in the chloroplast. This glycolate, which is 2-carbon compound from chloroplast, comes to peroxisomes, takes one more molecule of oxygen. Oxygen is excess only. It will take one more molecule of oxygen, and glycolate, after taking oxygen, it will become glyoxylate. Glycolate, after taking oxygen, it will become glyoxylate and hydrogen peroxide. So in the third reaction, which occurs in the peroxisomes, the glycolate, by taking oxygen, it will become glyoxylate and hydrogen peroxide. Now this glyoxylate, which is an acid, takes an amino group, becomes amino acid. So the fourth reaction is glyoxylate. By taking amino group, it becomes an amino acid. The simplest amino acid is glycine. So 1 and 2 reactions in the chloroplast, 3 and 4 reactions in the peroxisomes. So now we got an amino acid glycine. This amino acid glycine from peroxisomes, it will come to mitochondria. Now in mitochondria, 2 glycines will combine. 2 glycines will combine. From 2 glycines, 1 amino group out, 1 carbon dioxide group out. 1 amino group also will go out. Write down 1 amino group also goes out. And this amino group only can be used here. 1 carbon dioxide comes out. So we are telling carbon dioxide is released in photorespiration. And then, so if 2 glycines are combining, from which 1 amino group and 1 carbon dioxide is removed out, and then NAD is getting converted to NADH2, then both those structures will combine to make another amino acid called serine. So in mitochondria, glycine gets converted to serine. This serine from mitochondria goes back to peroxisomes. In peroxisomes, from serine, amino group comes out. Means amino acid, if you remove amino group, it becomes an acid. The acid is hydroxypyruvic acid. The acid is hydroxypyruvic acid. This hydroxypyruvic acid, it gets converted to glycerate. The hydroxypyruvic acid acid, it gets converted to glycerate. How it will get converted to glycerate means you have to remove this group, right? So that means NADH2 will come, gets converted to NAD. Here you made energy, that energy you used up again. NAD got reduced to NADH2. This is an energy source, but this energy source is used up again. So here we are telling NADH2 gets converted to NAD. Then hydroxypyruvate gets converted to glycerate. This glycerate goes back to the chloroplast, and this glycerate, it takes one ATP molecule and becomes phosphoglycerate. Phosphoglycerate is a starting compound. It's a starting product of the C3 cycle. It will start C3 cycle. So this 3-phosphoglyceric acid will start C3 cycle. This is the whole sequence of photorespiration. Who demonstrated it? It was demonstrated by Decker and Tayo in tobacco plant. Who told that there is a concept of photorespiration? Warburg told that green algae in the presence of more oxygen, it will not do photosynthesis. It will stop photosynthesis. Then it occurs in 3 organelles, right? It occurs in 3 organelles. It is because of Rubisco, which is a dual enzyme. It has oxygenase activity and it has carboxylase activity. If the CO2 concentration is more, it will do carboxylation. If the oxygen concentration is more, it will do oxygenation. If it is doing oxygenation, that means it's not doing photosynthesis. Stopping photosynthesis, doing respiration is called photorespiration. The significance of photorespiration in the biological systems is not yet understood. That's what the statement which is given in NCERT. The function of photorespiration is not yet understood. But yeah, photorespiration is decreasing the yield, is stopping the photosynthesis. That's why C3 plants will give less yield. C4 plants, since they are not wasting, they will give more yield. So with this, we finished photorespiration also. Take a screenshot, then we'll talk about the last topic, which is factors affecting photosynthesis. Now let us start about factors affecting photosynthesis. Now photosynthesis is affected by several factors. They can be internal factors or they can be external factors. Internal factors means they are the factors which are present inside the plant, right? So what are the factors which are present inside the plant? Internal carbon dioxide concentration, number of stomata, number of mesophyll cells, number of chloroplast, number of leaves, okay, water status. All these are the internal factors. Coming to external factors, what are external factors? Environmental factors means how much light is there, how much carbon dioxide is there, what is the temperature, what is the water concentration outside. All of them comes under external factors. So factors affect the rate of photosynthesis. If all the factors are there, then only the plant can do photosynthesis. If any one factor is deficient, then the plant cannot do photosynthesis, right? So factors affecting photosynthesis, we say that photosynthesis is under the influence of external factor also, internal factor also. External factors means environmental factors. Internal factors means they are plant factors. What factors comes under internal factors means number of leaves. If the leaves are more, photosynthesis is more. If the size of the leaf is more, photosynthesis is more. If the age of the leaf is more, means it's about to drop down, the green color is not there, then the photosynthesis is less. So plant internal factors include number of leaves, size of leaf, age of leaf, orientation of the leaf. If the leaf is well exposed to sunlight, photosynthesis will be more. If the leaf is rolled off, the area is less, photosynthesis rate will be less. Number of chloroplasts. See, if the chloroplasts are more, obviously pigments will be more, photosynthesis will be more. Amount of chlorophyll, pigments are more, absorption will be more, photosynthesis will be more. Internal carbon dioxide concentration in C4 plants, internal carbon dioxide they can increase to maintain the Rubisco activity. So all these are internal factors. Now again, the internal factors depends upon the genetic makeup, right? So if the plant genes are good, if they are strong, then all the internal factors will be good only. So the internal factors depend on the genetic makeup, genetic predisposition, and of the growth of the plant also. If the plant is growing nicely, then it will have all these things. Number of leaves will be more, shape of the leaf will be more, number of chloroplasts will be more, amount of chlorophyll will be more, right? So plant factors means all these factors depend on the genetic makeup and the growth of the plant. Coming to the external factors, there are 4 external factors. One is light. If light is not there, photosynthesis it cannot do. Temperature also is important because enzymes are there. Enzymes will work under optimal temperature. Carbon dioxide concentration. Carbon dioxide is a raw material which is reduced to carbohydrate. Then water. Carbon dioxide and water are the substrates for photosynthesis. External factors, 4 children. First one is light, second one is temperature, third one is CO2 concentration, fourth one is water, right? Now when so many factors are controlling, external factors and internal factors, when they're controlling photosynthesis, now all of them act simultaneously to affect the rate of photosynthesis. But among 4, usually only one factor will be limiting, which will reduce the rate of photosynthesis. For example, biology, physics and chemistry, 3 subjects will decide your NEET score. In that, it is physics which is the toughest one, which will decide like whether we will get a good college or not. Bio, it's easy when you prepare well. It's not at all difficult to score 360, trust me. Okay. So now here we are telling, when these many factors are there, let it be those many factors. All the factors are acting simultaneously, but there will be one factor. There will be one factor which will limit the rate of photosynthesis. And who told this? In the year 1905, Blackman proposed a law which is called as law of minimal factors. Blackman proposed a law which is called as law of minimal factors, which tells that when there are so many factors which are affecting the rate of photosynthesis, now if the rate of photosynthesis is less, whom to blame? Water to blame, carbon dioxide to blame, light to blame, temperature to blame. Check which concentration is below levels, blame it, right? Which concentration is below the required concentration? Because of this low concentration, photosynthesis is not happening. Who told this? In 1905, Blackman told it. It's a modification of Liebig's law of minimal factors with mineral nutrition. Now, when we have to talk about external factors, internal factors we can't do anything, right? They're there in the plant, we can't do anything. External factors we can control. Carbon dioxide we can control, water we can control. Let us talk about the external factors. Under that, the first factor which is given in NCERT is light. Light is very much important, right? Light is the only source of energy through which the plants will do photosynthesis. They'll convert the photon energy into chemical energy. Now we will discuss light under 3 side headings. What is the light quality? What is the light intensity? And about light duration, right? I wrote it here. Light we will discuss under 3 things: light intensity, light quality and light duration. So under light intensity, we can tell that like at low light intensity, plant will not respond. At low light intensity, plant will not respond. Now at optimal light intensity, photosynthesis will be good. But at high light intensity, if you take high light intensity, at high light intensity, the chlorophyll pigment get destroyed, right? The chlorophyll pigment get destroyed. What is that concept called? Photooxidation. It is called photooxidation. It is called solarization. At very high light intensity, the pigment is destroyed. When the pigment is destroyed, no one can absorb the light, then the photosynthesis goes down. So, under light intensity, we are telling at low light intensity, photosynthesis only will not happen. At moderate light intensity, photosynthesis is going good. The rate is good. At very high light intensity, then the pigments are destroyed. The concept is called photooxidation or solarization. Coming to light quality, the range of spectrum what we see is VIBGYOR, we know this. Under this, we also know that plant absorbs good at blue wavelength and at red wavelength. So, which light quality the photosynthetic rate is high means at blue and red. At which light the photosynthetic efficiency is least means at green it is least. And at blue and red it is maximum. This is about light quality. Next one is light duration. So whether continuous light duration will increase the photosynthesis or discontinuous duration will do photosynthesis. If I teach you continuously from morning to night, is it efficient? Or if I teach you like 1 hour in the morning, 1 hour in the afternoon, 1 hour in the evening, so that it will get into your brain, you can understand it peacefully. The same manner here also, continuous light will lead to stress. Intermittent light means light and then rest, again light, again rest. So we can tell that intermittent light will increase the rate of photosynthesis. So we finish the first factor, that is light. So light intensity, moderate light intensity, neither low nor high. Light quality, blue and red. Light duration, intermittent duration, not continuous duration. Next, coming to the second factor, which is carbon dioxide. Carbon dioxide is the raw material, but the amount of carbon dioxide which is present in the atmosphere is very less. How much it is? 0.0 0.003. It is 0.003 to 0.004. It is very less. Now if you try to increase the carbon dioxide concentration from 0.003 to 0.004 to 0.05, the rate of photosynthesis also will increase. The rate of photosynthesis also will increase. And again we tell that C3 plants require more carbon dioxide. So in C3 plants the carbon dioxide compensation point is high. How much? It is 450 micrograms per liter. We saw it in the differences between C3 and C4 plants. But C4 plants, they get saturated at low levels of carbon dioxide. What is that? 360 micrograms per liter. C3 plants respond to high concentrations. How much? 450. So that's why the productivity also will be more. More the carbon dioxide, more the carbohydrate. So greenhouse crops like tomato and bell pepper, you take, they are C3 plants. So if you give more carbon dioxide, they'll do more photosynthesis, they'll grow more. So people are commercially exploiting this bell pepper and tomatoes, and they're growing in greenhouse rooms, so that more the carbon dioxide they're exposing, then more the photosynthesis and higher the yield in less span of time. So children, this is about carbon dioxide. Carbon dioxide is a major limiting factor. Light is not at all a limiting factor. Light is not at all a limiting factor, because sunlight is having 100%, and what plant is using is 1 to 10%, maximum 10%, minimum 1%. 90% is extra with the sun. So sunlight can never become a limiting factor. Carbon dioxide is a major limiting factor. CO2 is a major limiting factor, has been asked in the NEET examination once. Now let's go to the third factor, that is temperature. Now when we talk about temperature, we know photosynthesis is having light phase and dark phase. In light phase, ATP is made. In dark phase, like it is C3 cycle, C4 cycle, each step is mediated by many enzymes. So the dark phase reactions are enzyme mediated. There the enzymes are sensitive to temperature. So we can tell dark reaction, being enzymatic, is more controlled by temperature. Light reaction is also affected at high temperatures, but when compared to light, dark reactions are more affected. And we know the optimal temperature for C3 plants is 20 to 25, and for C4 plants it is 30 to 45. So C4 plants respond to higher temperatures. Higher the temperature, higher the yield. Tropical plants will respond to higher temperatures. Tropical plants have higher optimal temperatures than temperate plants. C3 plants are temperate plants, C4 plants are tropical plants. So this is about temperature as a factor which controls the rate of photosynthesis. The last factor is water. Now carbon dioxide plus water gives rise to carbohydrate. Means water is a reactant also. Carbon dioxide is one reactant, water is another reactant. So water is reactant also in the light reaction, right? So the effect of water as a factor is more. The effect of water as a factor is more rather than on photosynthesis, right? So what is the effect of water on the plant is more rather than carbon dioxide plus water giving us to carbohydrate. Now if you don't water the plant, plant will go to stress. And high stress, plant will not do transpiration. If it should not do transpiration, it will close the stomata. If it closes the stomata, then from how it will take carbon dioxide? If carbon dioxide it is not taking, then it cannot do photosynthesis. So if you don't water the plant will not do transpiration, so that it will not open stomata, so that it cannot do photosynthesis. Not only that, if you don't water the plant, the leaves will wilt. The leaves will shrink, they will wilt. When they are wilting, they cannot take the sunlight. When they are not able to take the sunlight, metabolism, energy will not be made, metabolism will go down. So the four factors we discussed, children. First one is the light, second one is the carbon dioxide, which is a major limiting factor, third one is the temperature, and the fourth one is the water. And then they have given this diagram and they're telling the graph for light intensity. So this is light intensity we are talking, right? At low light intensity, the rate is less. As you are increasing the light intensity, the photosynthesis is also increasing. See, as we are increasing the light intensity, the rate of photosynthesis is also increasing. But after one point, it got saturated. After one point, it got saturated. You are increasing the light. You are increasing the light, but the rate of photosynthesis got saturated and it's getting maintained. Why? You are increasing the light, but the other factors will become limited, right? Either carbon dioxide or water or temperature becomes limited. So because of the other limiting factors, the rate of photosynthesis will get saturated. So as you increase the light, the rate of photosynthesis increases. So with this, children, I think we completed the chapter photosynthesis. So if you like the content, like, share, subscribe to my channel, share it with your friends. Any doubts, post them in the comment box. Thank you.